Very good morning, sir.
Hi, good morning.
Good morning, sir. Good morning. Sir.
Can you hear me?
Can you hear me?
Yeah, sir. Audible, sir.
Hello, can you hear me?
Yeah, sir. Audible, sir.
Okay. I just want to make sure that you can hear me. Right? Okay.
Yeah, sir.
Now it's fine, right? Clear?
Yeah, sir.
Okay.
Very good morning to respected Principal Madam, Dr. K. Padmalata, convener of this 2 days national webinar on recent horizons in experimental pharmacology and molecular biology. And today's resource person, Dr. Sabrina Garu, Dr. Tripathi Garu, and Dr. M. Wani Garu, all faculty members and students who participated in this 2 days national webinar. On behalf of faculty and IQSE, Vijaya Institute of Pharmaceutical Sciences for Women, it's my pleasure to welcome you, sir, to this national webinar hosted by the Department of Pharmacology and Department of Pharmacology. For the convenience of participants and resource persons, this 2 days conference conducting in virtual mode. Thank you all for your active participation in this national webinar, and your active participation will add more value to this conference. Thank you all. Now I request our beloved Principal Madam, Dr. K. Padmalata, to address the opening remarks of today's virtual conference. Now it's hand over to our beloved Principal Madam.
Thank you, Dr. Venkateshwara Garu.
Thank you, Madam.
Myself, Principal of this Vijaya Institute of Pharmaceutical Sciences for Women. I welcome all the registered participants, speakers, my students, staff, and all well-wishers to this virtual conference scheduled for today and tomorrow on the theme Recent Horizons in Experimental Pharmacology and Molecular Biology. This conference is organized by Department of Pharmacology and Department of Pharmacognosy of this institution, and I congratulate the staff of these two departments for their enthusiasm in choosing the today's and tomorrow's speakers and making this conference to happen. I'm very much thankful to Dr. Sabrina from CSIR, IICT, Dr. S. Tripathi from Anusandhan University, Bhubaneswar, Dr. M. Wani, our faculty, Department of Pharmacognosy, Dr. A. Sai Balaji, a scientist, again from IICT, Hyderabad, and Mr. Karthikeya Reddy from Cognizant, and Mr. Jairam Reddy, our faculty from the Department of Pharmacology, who have come forward to deliver talks on the present theme, Recent Horizons in Experimental Pharmacology and Molecular Biology, in this 2 days program. So I hope the participants and my students will get a valuable information relevant to research for their future plannings. And even the students of MFAM, those who have joined this program, they will have an ideology of how to mold their future in the research field. So with these few words, I congratulate the organizers. I thank the speakers, and I wish and bless all the participants for their valuable time and betterment through this conference. Thank you, one and all.
Thank you, madam, for your valuable welcome address of today's webinar. Now, I would like to invite Dr. M. Wani Garu to address the welcome note of today's international conference.
Good morning, respected Principal Madam, dignitaries and speakers of today's webinar and delegates. A very good morning to you all. I would like to welcome you all to today's virtual national two-day webinar on recent horizons in experimental pharmacology and molecular biology, organized by Department of Pharmacology and Pharmacognosy, Vijaya Institute of Pharmaceutical Sciences for Women. I'm sure that you already have noticed our program is filled with interesting and prominent speakers from different parts of our nation. This stands out a great interest towards this program. We know all that the health of society is the key, and our today's program is intended to focus on new insights, novel developments, current challenges, recent advances, and future perspectives in experimental pharmacology, drug discovery, and molecular biology. In today's scenario, we really need topics like this, and research studies at molecular level are really important to exactly know the disease mechanism, interactions between chemical mediators and cell components in cell environment to target a particular disease. This is very much helpful in drug discovery and development and disease diagnosis, so which are essential in therapeutics. I expect the talks of today's program will give more insights and a path forward. And I hope from all the speakers we'll get enlightenment on this topic. And we extend this topic towards natural products also, whose exact specific mechanisms are not known. Therefore, the studies at molecular level on natural compounds will provide new leads towards relevant pathophysiological targets. So we are grateful to all the speakers. They have all accepted our invitation, and I wish a grand success. Thank you for giving me this opportunity. Now I hand over the session to Madam Sunita Garu. Madam Sunita Garu will introduce Dr. Sabrina, sir.
Myself R. Sunita, Associate Professor in Pharmacognosy and Phytochemistry, Vijaya Institute of Pharmaceutical Sciences. Good morning, everyone. It's a great privilege to introduce Dr. Sabrina Chilka Garu, Scientist, Ramalinga Swamy Fellow, Department of Applied Biology, IICT Hyderabad. Sir completed his bachelor's degree from Nagarjuna University during the year 99. Later, 2001. So completed Masters in Biotechnology from Pondicherry University. Later, he worked as research associate at the Center for Cellular and Molecular Biology at Hyderabad. There, he worked on characterization of chromatin insulator elements, sequence analysis, and identification of highly conserved non-coding DNA elements in the mouse. He did his PhD and postdoc program in molecular biology in the Institute of Molecular Biology at University of Zurich, Switzerland. There he worked on study of transcription regulation, enhancer promoter specificity using Drosophila melanogaster genetics. Later, from 2009 to 13, Sir had one more postdoctoral program in the lab of Dr. Marie-Noël Préaly, Institute of Jacques Monod at University of Paris, France. Later, he worked as research associate in Epigenetics Lab, Institute of Cancer Sciences at the University of Glasgow, UK. And during the year 2017 to 19, he worked as research associate at Institute of Infection, Immunity and Inflammation at University of Glasgow, UK. Later, from 2020 to present, Sir worked as Ramalinga Fellow at Applied Biology, CSR ISCT Hyderabad. There he worked on epigenetics and gene regulatory mechanisms of inflammation, mRNA vaccine platform development at ISCT. He received many awards and recognitions. During his bachelor's degree itself, Sir received Geology Department Memorial Award and Swarnalatha Memorial Award for the excellence academic excellence. Later, Sir selected for the Zurich Molecular Life Sciences PhD program, Switzerland. Sir qualified CSIR UGC NET exam in 2002. Eligible exam at Indian universities. Later, Sir selected for the Ramalinga Re-Entry Fellowship, DBT, Government of India, hosted at CSR ISCT, Hyderabad. Sir attended many meetings and completed various courses. He completed European Molecular Biology Organization practical course during 2010. Sir delivered an oral presentation on chromatin replication and chromosomal stability at UPCAM conference, Stockholm, Sweden. Sir had given poster presentation on epigenetics in dialogue with the genome at UK. Sir has various publications in reputed international journals. Sir had submitted conference papers at Arthritis Rheumatoid. With this brief introduction, I welcome you, sir. It's our privilege to have you here to deliver a lecture on mechanisms and molecular techniques of inflammation and associated disorders. Sir, please.
Thank you, ma'am. Thank you for a very nice, kind introduction.
Thank you,
I
sir.
hope I'm able to, I'm audible, right? You can hear
Yes,
me?
yes, sir.
Okay. Thank you. It's very, very elaborate introduction. Okay, thank you very much. Can I share my screen? Yeah.
Yes, sir.
Okay. So you have to make me the presenter. Okay, I get it.
Yes, sir.
Okay. Can you see my slide presentation?
Yes, sir. I have given multiple sharing options, sir. Now we can share the video. Yeah, it's visible, sir.
Right. I'll stop the video so that it will better. Okay. 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 Hi. Good morning, everybody. I thank all the principal ma'am, Padmalaga Garu, and all the staff members of Vijay Institute of Pharmacology. Thanks for giving me the opportunity to give a guest lecture. I'm very happy and excited to be here, and I hope I will be of some use. Today's talk, I'll give you half general introduction, like the field and stuff, and rest at the end, I'll tell what I am doing as a researcher in the IISCT. Okay, as it was introduced, my name is Sabrina. I'm a Ramalinga Swami Fellow. It's a fellowship given to the researchers by Government of India, DBT, Department of Biotechnology. Okay, let me start. Today, I'm going to talk about mostly on inflammation. Okay, so please tell me if I'm not audible somehow because we don't know net whatever stuff. Okay.
Okay, sir.
Okay. Okay. It is running a little bit slow, but okay. Okay. Right. What is inflammation? Inflammation is a fundamental response of our immune system against any harmful stimuli, which is it can be microbial invasion. That means it could be virus, it could be bacteria, it could be fungus or parasite, and it could be toxic substances, or it could be damage. Let's say a cut happened on our skin, or we get some fever, a cold, or some bacterial infection can happen anywhere in the body, or some toxic substances. Then our body gets activated for inflammation. It will send a lot of cells of the immune system to the site of damage or to the site of inflammation, site of infection, and will start the process of fighting against the infectious agents or to cure that damage. Okay, so that involves, see, there's a damage happens, then the local cells alarm the body. Okay, now the nearby blood vessels, they get the signal and they will get loosened so that the lot of blood cells will enter here. Immune cells will go and enter there and start the process of fighting. So it is our defense system. It's an important system for our body. Okay, so that's the inflammation. Inflammation is highly regarded as mainly four signals. One is it will generate heat at the site of infection, redness and swelling, pain. We all know that. And also recently it was joined like a loss of function because you will not, if there's some damage happen, you will not be using that place very well. So these are the main symptoms of the inflammation. Okay, I told you. So when some damage or some parasite or some bacteria viral infection happens, I told you the immune system gets activated and it will send a lot of cells. So what are these cells? Let's see. These are our cells of our immune system. So mainly it will be regarded can be two parts. One is myeloid lineage, another is lymphoid lineage. So here you see the lymphoid lineage, and here you see the myeloid lineage. Okay, so these are the very, very important cells of our immune system. They all come from one progenitor, which is hematopoietic stem cells, which reside in the bone marrow. From bone marrow, some progenitors will come. The lymphoid progenitor, that cell will derive different B cells, T cells, and natural killers. Again, in T cells, you have helper T cells, cytotoxic. They have different functions. For example, B cells are for producing the antibodies. You all know that antibodies are produced against different pathogens when they get invaded in our body, right? And on the other side, we have a myeloid lineage where you have neutrophils, eosinophils, mast cells, blastocysts, and importantly, monocytes, which again give rise to dendritic cells and macrophages. So what are they? Why we have to look at them? See, just remember these part, the lower myeloid part is like a non-selective. They are kind of the police going around all over our body mainly in the blood, but they can go to anywhere in the body, and they have a watchful eye. They are kind of the guys which are looking for anytime anything, any invasion happen. Once any invasion, what kind of invasion? Their job is to first line of defense. They will attack those. If it is bacteria, if it is virus, anything, they will first find it, and they are the ones who say, "Okay, I found something. Please." So they start the fight first, and also they call later the adaptive immunity. This immunity by T cells and B cells is very specific. So one particular bacteria comes, so they will make a defense for that particular bacteria. If there is a virus, then that particular viral defense it has to do. So this is innate immunity, that is adaptive immunity. More specific, and the bottom ones are not specific. They are very, very general stuff. Okay. So just remember, these are also are the dendritic and macrophages are the antigen presenting cells. See, neutrophils, monocytes, which derived dendritics and macrophages, they not only defend the thing, they also present the whatever the parts of that pathogen to these cells. They also call them, and then they will ask, "See, I found something here, a foreign body, come and then start your part of the job." Okay, so all the things, all these cells are involved in the inflammation. They all trying to work initially to defend the exogenous pathogen, and then they also do at the later stage, they also do another important part of the job, which is they also reduce the inflammation. They want just themselves, they organize themselves, they communicate themselves to make the inflammation stop, and then also repair the process. Okay, so you all know that we get some cold. It will stay for like, let's say, maximum 3-4 days, and then it goes away. Similarly, some bacterial infection can come, some fever comes on 3-4 days or 1 week, it will go away. So that is very acute. That means when the pathogen infection, our inflammation rises to an extent where it will fight, and then remove the bacteria, remove the viral things, the foreign substances, and then it will go into the repair phase. Here it is a destructive. That's why it is inflammation. Flame is fire actually. So they will, in process of making the defense, they damage the tissue. That's how they work. And later then they subside. When the pathogen subsides, then it says, okay, it is done. Now we don't need any more the inflammation. Let it cool off. So this resolution phase, they do the repair, the damage caused by the inflammation. But many of the diseases we have in the world, they are due to the cause of long inflammation, chronic inflammation. So they are not just coming and going. Instead, they are keep on maintained. So what happens like that? Then the damage is continuously happening. That causes our severe pain in our body, muscle pain. It could be muscle chronic inflammation or rheumatoid arthritis, for example. So here I give you chronic inflammation is inflammation for long time. Okay, several times you can ask me why we have to have inflammation for long time. Let's say I just told you, if a bacteria comes and then it fights and it clears off, then you don't need inflammation anymore, right? But what if the bacteria, the system cannot remove the bacteria? The immune system is finding it so difficult to remove. Then we will have long inflammation come. It will maintain. Then the pathogen is there, inflammation is maintained. But here this is interesting. Many of the diseases we have, I'm showing you here, many of them are autoimmune diseases. At one point, we don't know the reason yet how the chronic inflammation comes. Somehow, system does not subside. I told you at the end of the inflammation, it has to go into a phase where it has to do anti-inflammation, stop the inflammation signal. Stop the inflammation signals are not coming anymore. That's why the inflammation keep on continuing, and it is damaging our own body parts, causing several diseases. You can see here the list of diseases which are due to the chronic inflammation. Most of them are autoimmune. That means autoimmune means they are destroying our own body. Somehow they got a wrong information. They're targeting our own tissue, and they're thinking that is foreign and trying to do the inflammation, unnecessary inflammation. So how we can stop those system is obviously to control that fire. We have to control that fire. Okay. Okay. So before going to that part, I showed you, right, several of the antigen presenting cells or immune cells. They are the watchdogs of our immune system. They're always trying to keep an eye on our body system. So these cells, how can they identify which pathogen came, what came? Interestingly, these general immune cells, which is macrophages, dendritic cells, natural cells, see on their cell surfaces, they have peculiar receptors. They are called toll-like receptors. So that's why these toll-like receptors, there are several kinds. At least you see TLR 11, 12, at least 12 kinds you can find, and each one is specified for one particular pathogen. For example, see here, TLR4, I'm highlighting, it is the recognizing LPS. This is a bacterial cell wall lipopolysaccharide. It's a polysaccharide. So whenever the cell sees a bacteria, the bacteria coat has the lipopolysaccharide, and then this cell signals this. Okay, look at that, TLR4 signaling activated. So it is activates downstream from proteins, and those goes further down, and then you have the production of several key proteins which are doing calling and also making themselves more number, stuff like that. Okay, this is TLR4 for LPS, which is bacterial. Likewise, we have bacterial, another TLR1 to 6 recognizes bacterial lipopeptides, and another one is bacterial flagellin, TLR5, and also in the endosomes, you have double-stranded RNA recognizing TLR3 recognizes double-stranded RNA, TLR7 and 8 recognizes single-stranded. Why single-stranded, double-stranded? Because several viruses are double-stranded RNA and single-stranded RNA. So whenever they ingest, let's say cell ingest or infected the virus, they end up in the endosome. In the endosome, they release their single-stranded RNAs, and then our cell recognizes. It's like a watch eye. The single-stranded RNA came, then it will activate TLR7, and then further downstream signaling pathways are activated in the cell. Similarly, we have double-stranded DNA for bacterial infection, TLR9, for example. Okay, so what you need to understand is that our cells are very well equipped to identify whatever the kind of pathogen we have. Okay, that's okay to recognize, but in the what about in the chronic inflammation? There is no pathogen there around. But how? That is the problem. These pathways are somehow constitutively active. That means without no pathogen, they are still firing and then causing the inflammation. Okay, one signaling pathway you should remember for any inflammation, right? Inflammation means you have to talk about nuclear factor kappa B, which is NF kappa B signaling pathway. So I just told you TLR, or it could be any other cytokines like TNF alpha, several other stimuli. These are all inflammatory stimuli. When they got the stimuli, the cell sends signal to these proteins called IKK complex. This IKK complex, there is a protein called IKK beta, which gets phosphorylated upon the stimuli. And what it does is, when this gets phosphorylated, it gets activated. It is a kinase. That means it adds phosphate group. So to whom it adds phosphate? It phosphates to this I kappa B alpha. This I kappa B protein is phosphorylated. What is so important about it? Look, this is the P65, right? This blue P65, P50 complex. These two are the part of NF kappa B. They are the important. This protein I kappa B is binding them and stopping them to enter into the nucleus in the normal condition. But when stimuli came, what happens? This complex gets activated, that gets phosphorylated. Look what happens when it gets phosphorylated. It is get a signal, the cell will get a signal to get ubiquitin. Ubiquitin is a small tiny protein moiety which is added several times. So what does it do? Whenever any protein gets a polyubiquitination chain, that will get degraded. It's like shredded and degraded. So this protein is not longer anymore present in the cytoplasm. So then what happens? These two are free to go. There they get phosphorylated, and then they enter into the nucleus. This is where we have the genes, right? You all know that DNA has so many genes, and all of them are doing coding for some proteins. In this case of inflammation, this NF-kappa B is a transcription factor, will go and bind to some parts of the genes and activate genes which are required for the firing, that is inflammation. So they are usually we call it as pro-inflammatory cytokines. Importantly, we call TNF alpha, IL-1 beta. These are the few examples, right? So you understand. So when there is a stimuli is there, the NF-kappa B, this is one of the important pathways, but we have several other pathways also. But ultimately what they do, they enter into the nucleus and activate the gene. You just have to understand the genes are dormant, they are not on yet, but they're waiting for the signal. That signal is this NF-kappa B. It goes inside, binds to the several promoters and activate the required protein. And what do they do? These are the important parts of the inflammation. So we have several cytokines. They are called cytokines, chemokines, and so on. So cytokines, they, as you see, we have acute. Few of the even acute is inflammation. Few of the cytokines are active. Whereas in chronic inflammation, this number increases. There is so much overactivated inflammation is going on. And anyway, these are the very important cytokines listed here. Among them is the TNF alpha, tumor necrosis factor alpha, mainly produced by macrophages, NK cells, and T cells. They and also IL-6 and IL-1 beta macrophages. So macrophages, monocytes are the major producers of these cytokines. What do? These are the small proteins which communicate with other cells. If a macrophage or monocyte release the cytokine, these cytokines, once released, they all the other cells. It's like calling, okay, you come here. Now you have to proliferate now. Now you start your function. That means then they themselves activate in their cell TNF alpha signaling pathway, and they again produce different kinds of cytokines. So production of cytokines, we just have to get one thing is that they are communicating each other. They're communicating, they're calling each other, and finally, altogether, they're trying to make a big war, thinking there is a pathogen present there. If it is a pathogen present there, it's fine. We are good. It is eating them. It is clearing them. But whereas there is no pathogen, then it's actually damaging our tissues. Okay, how we are treating for this inflammation? So this is a little bit. We will go inside the some drugs which are used. So mainly we start with the non-steroidal anti-inflammatory drugs. So as you all know, they are not steroids, right? They are chemicals which are mainly of the COX inhibitors. You know that they are aspirin, ibuprofen, indomethacin, right? You know all these common chemicals will everybody knows. So and there is a very selective inhibitor, colegionatory etorecoxib and paracoxib, right? And diclofenac, meloxicam. All these are COX inhibitors. What are these COX inhibitors? So did I talk about those? Where do they function? No, I haven't talked about that. So these are non-steroidal, and they're actually, you know what they're doing? They are here. Look at this signaling. They are producing COX-1, COX-2 enzymes, cyclooxygenase enzymes, which they produce prostaglandin. They are the lipid chemicals, lipid moieties, small lipid moieties, which function in inflammation, hemostasis, and protection of gastric. They are different functions. As you see, their PGE2, prostaglandin E2, and so on, all these ones are what they're doing. They are helping inflammation. They are making a fire actually. You should think that. So that is produced by COX-1, COX-2 from the arachidonic acid, which is from the membrane lipid. It makes the prostaglandin, and this is produced by many of the cells. So since they are the general inflammation gainers, so they have devised non-steroidal drugs. They design those drugs, they bind to the active sites of these COX-1 and COX-2, and they prevent the formation of prostaglandins. When the prostaglandins production is reduced, then we have inflammation and pain is reduced. Okay, so these are the non-steroidal. Most of them are COX-1, COX-2 inhibitors, and they are lipid-based. Just remember that. So the prostaglandin production is inhibited, and since they are prostaglandins are important for inflammation and pain and fever, all these things can be reduced by doing this. But these are not very specific to the inflammation which we talked about, right? So how to stop those ones? So those ones are, well, still there is another class which is called corticosteroid, or generally called steroidal drugs. You would be definitely heard about dexamethasone, cortisone, right? Prednisolone is one of the commonly used. Commonly means commonly used for certain diseases only. You cannot use steroids just like that for every fever or every pain. You cannot do that. Why? Because they are very hot chemicals. The corticosteroids, when they enter into the cell, the cytoplasm, they bind to the glucocorticoid receptor, which will stop the NF-kappa B complex. I told you, right? NF-kappa B is very important, activating several cytokine genes, which are important for inflammation. So it will repress, transrepress. It will go and bind to the NF-kappa B complex, and it will stop its binding and stop its activation. They also found to act by binding to the DNA and also some activation of the anti-inflammatory gene, or actually they repress also some genes directly. See, that is a DNA binding, and this is a NF-kappa B. Whatever it is, see, corticosteroids are very, very harsh. You can only give, because they're very strong chemical substances, and they shut off the immune system. That is very dangerous for a common use, because steroidal use can bring more infections can come, because our immune system sleeps, then there is more chances that more other severe infections can come to the body. Anyway, but we have to give to some people. Why? Because steroids are used for the certain rheumatologic inflammatory, where normal chemicals or the non-steroidals are not working. We have to give them, otherwise their muscles are there, the joints are getting damaged, they're getting eaten up. So that cell damage processes by inflammation, the massive non-steroidal drugs does not solve. We have to stop them. In those cases only steroidal drugs are given, but just be careful that you can go and see the literature. Steroidal drugs comes with several defects. On the long-term use, muscle weakness, kidney damage, liver damage can happen. So one should be the doctors will very carefully low doses they try and give them. But some conditions we have to give, because we don't have any other choice. We have to stop the damage to their body. That's why they have to give and suppress the immune system. Okay, so this is another class. This is the latest class of treatment being given again to some particular diseases. This is what here I told you, the cytokines are very important in communicating, in making the inflammation. Yes, sir. So one way is to stop them, stop the production of the cytokines. Now another way here is you stop those ones which are already produced by using monoclonal antibody. So antibody technology is in our body, B cells produce some antibodies against some particular pathogen, right? So then whenever the pathogen comes in, the antibody will bind and then stop its production, and it makes the cell to be eaten by some other cell. But that is the technology the antibody is working. But here the antibodies are engineered against the protein, TNF alpha protein. So what happens? They will inject this monoclonal antibodies for against the TNF. So when these antibodies are injected, what they do? They will go and recognize the TNF and block them. At least they will prevent the TNF go and do its function. Similarly, they have done IL-1, IL-6 and stuff. So these all of them are in use currently. FDA has approved them. This is the monoclonal antibody therapies or immunotherapy. We call them as biological therapy because all these are all biological agents. Proteins or biosimilars are the protein. They are not chemicals. They are not chemical substances to block the TNF. They are the proteins which are against that cytokines. So the proteins against the proteins. Whenever they see these TNF, they block the function. So that like that. So when they're blocked, you can stop the function. That means it's not able to go and bind to the place where it's supposed to bind. So inflammation is reduced. Similar fashion, you have to imagine. So I just told you there are several of these are the different monoclonal antibodies currently approved, and it is people have been using. But again, you have to be careful that no, we cannot just inject all these monoclonal antibodies just like that, right? They are again comes with a very strict scenarios only where you can give, because we have to try non-steroidals and steroid, maybe steroidal or some DMARS or any other drugs. They all fail. They all cannot stop the degradation or whatever the damage causing to the body due to this over inflammation. Then only doctors prescribe them. All these antibody therapies. You see, etanercept and adalimumab, all these MAB is a monoclonal antibody. Okay, infliximab against TNF, golimumab, and IL-1, IL-6, tocilizumab. Right. All these are biological therapies. So they are antibody therapies. Okay. So again, they are again stopping those very important cytokines which are doing the inflammation. Right. So these are the techniques. These are the different methods we are treating therapeutical ways: non-steroidal, steroidal, and we have a biological therapy. Okay. So before jumping into my work, let me give just two techniques, small techniques, general techniques, so that you will understand when I talk about. See, whenever I said gene activation, right? So when I said TNF alpha, NF kappa B will go into the nucleus and activate genes. So how do you know that the genes are activated? Whenever a gene activated, that means that kind of protein has to be produced at the later point. But before to the protein, the TNF alpha, let's say example, TNF alpha protein has to come. So NF kappa B goes and activates the transcription. Transcription is meaning it makes the mRNA from the DNA. So that mRNA we want to quantify. We cannot quantify directly mRNA by PCR. Polymerase chain reaction is a technique. We cannot directly use the RNA. We have to make the RNA to the DNA, which is called reverse transcription. So we take the RNA, total RNA from the cell, and we make a DNA strand which is complementary to the mRNA by using the reverse transcriptase enzyme. Once we got the DNA, which is, we call this as complementary DNA, that is cDNA synthesis. Once the cDNA synthesis is done, we'll use this as a template to make qPCR, quantitative reverse transcriptase PCR. What happens in that PCR, we need to give specific primers. That is, if I want to detect TNF alpha, I have to design TNF alpha sequence specific primers. So using those primers, we run several cycles using a fluorescent dice. So that fluorescent dice will detect the live, how much DNA content it is increasing. See, more the RNA we have, that means more the copies of TNF alpha is there, it will take less time so that it will amplify a lot. If you have very few copies, then it takes long time. This is the cycle actually. CT, we call it CT value, cycle number. So it takes long time so that it will get produced in a good amount so that the machine detects particular threshold. So using the quantitative PCR, we can identify which gene is active, which gene is not active. Right? We always have a housekeeping gene which doesn't change, and we have a induced genes which will change. We will normalize the housekeeping gene, that is the gene which doesn't change, with the genes which we are interested in, and then we quantify the gene expression. right? So this is one of the major techniques we use in the lab for gene expression analysis. That is gene activation. Gene activation means mRNA is coming. Okay, just remember. The another method is everybody knows is the ELISA, right? In the ELISA assay, what we do, we are detecting the protein. Previous gene expression qPCR is detecting the RNA. Here we are detecting the protein. How we do that? We take a 96 well plate and we coat the bottom with the antibody of interest. If I want to study IL-6, then I'll coat the antibody for IL-6. If I want TNF alpha, I'll coat with the TNF alpha. And then the coated one, I add my sample. So if my sample has, let's say, TNF alpha, that antibody recognizes and it will bind. And then after washing, I will put another TNF, anti-TNF alpha antibody, which is biotin labeled, and then avidin. After washing, each step after washing, what we do is avidin and it's a HRP. So it's an enzyme which makes the color reaction at the end. So the level of binding, the more abundancy will make the more secondary this antibody, which has the will be binding. So more color reaction. So by using the color in this right ELISA reader, you can quantify the how much TNF alpha or how much IL-6, or in that case, how much any protein can be tested by this assay. So by using the simple coloring assay in the 96 also, we have a standard curve fitting the standard curve against your unknown sample. We can plot and then quantify. Right. So I hope you all got these are the two techniques we normally use in our lab to see the gene expression and ELISA with the protein expression. There is also another one, Western blot, to look at the any protein on the gel. Okay. So I hope I have time, right? I'll finish in 10 minutes. Okay. So this is quickly a few slides about my work, what we are doing at IISCT. So we are doing small molecule epigenetic drug screening for identification of anti-inflammatory compounds. So for this, what is epigenetics? I have to introduce to you because this is my field and I thought it would be nice to know. I told you gene activation is very important. See, all our cells have all the genes required. So if you take from neurons to the muscles or any immune cells, all our cells have the DNA, same number of copies of genes. All the genes are there in every cell. But a neuron cell will only produce neuronal genes. A muscle cell will produce only muscle genes. For example, in our case, in the case of inflammation, only inflammatory genes have to be produced, not other genes. How this is possible? It's possible because of the chromatin higher order structure. How does it happen? We have a DNA, double-stranded DNA, which is wrapped around a histone protein. There are some 8 histone proteins which are wrapped and forms beads, like beads on a string model. And this structure again forms a higher order, like they again coil themselves, make a higher order structure. So just remember, DNA is not just like that. What we see, DNA in the pictures, is not like that. It is actually wrapped around several proteins, mainly the histones, and makes a higher order structure. Why we have to important? This is where it is important. See, when a cell want to off the gene, it will make the chromatin very compact. It will not allow any other transcription factor like TNF kappa B. It will not allow them, so that it is gene off stage. When it has to switch on, it will loosen up the chromatin and makes it open. Okay. So how this is possible is because I just told you that beads on a string, right? That each individual, this is called nucleosome. This is where the entire thing is happening. Actually, if you look closely, that nucleosome has all the histone proteins, tails are protruded out. These are internal tails. And they are nothing but there's some amino acids, right? They are amino acids. These amino acids are targeted for acetylation or phosphorylation or methylation. Just small moieties, they will add or remove. Basing on the type of modification, the chromatin can become compact off stage or on stage. So it depends on the how this regulation is happening. Okay, what is interesting for us? See, here I tell you. For example, let's say TNF alpha gene is off in the normal condition. Now we have a stimulation came, like inflammatory stimuli comes. In our case, we add LPS. What happens? LPS happens. NF kappa B get activated. It goes inside the cell. Now, what does it do? It will bind to the promoter and activate the TNF alpha gene. That means TNF alpha inflammatory cytokine K. Well, can NF kappa B can do all the opening by itself? No, it has to take the help of other proteins which are opening. They are called epigenetic factors. So, in order to open the chromatin, it needs to take. It's well known that it talks to the different factors. Okay, what if we stop those epigenetic factors using small molecule inhibitors and stop the opening? So even though enough kappa P goes inside, it cannot open because we blocked those XYZ, right? Other epigenetic factors. In that way, what can we achieve? If we can achieve the TNF alpha activation inhibition, that means we are reducing the TNF alpha. This is one example I'm telling you. So it can be, this is what we want to do and this is what we do in the lab. So for that, we mainly focus on monocytes and macrophages. We use a cancer leukemia, which is a monocytic leukemia cells, the in vitro model, which is in the lab we grow in the petri dishes. We grow these monocytes and we work on them. We can in fact transform them into macrophages with using a PMA chemical. Okay, what we do, we grow these cells, these monocytes and macrophages, we give LPS. Why we give LPS? When we give LPS, this is a purified LPS coming from a bacteria. The cells think that there's a bacterial infection came, and then they start inflammation. How do we know that we start inflammation? We'll tell you in just the next slide. See here, when we add LPS, the cells get activated. When they're activated, then we isolate the RNA and we do the qPCR and then find the expression of these genes. That's how we know it, that they're activating the, for example, look, LPS at different concentrations. When we give to this THP1 monocytes, then TNF alpha transcription is increasing with the, this is IL-1 beta. Look at that. So mRNA is getting from 0 to 200 folds it increased. Here it's almost 100 folds it increased. And this is the time wise. It takes just 2 hours to activate the inflammation further, right? So it's very quick and very robust. So similarly, in the PMA differential macrophages, we have macrophage model also. We find the upregulation of the inflammatory cytokine. Okay, so what we do here is, once we made these macrophages, we add epigenetic library. See, because I am interested in epigenetic factors, right? I just told you all the chromatin opening factor inhibitor library is available commercially, which is like around 140 chemicals we have, and we treat all, like in the plate, we treat those cells with the epigenetic inhibitor, and then we add LPS to that. So you tell me, when, if I did not add any of them, the LPS is supposed to increase the inflammation. But if any of those chemicals inhibit, then LPS will not lead to the activation of TNF alpha. So this is exactly what we are testing. We are just doing several compounds. We add these compounds and we add LPS and asking any of them are decreasing or increasing, right? So you see here, just they're all the same, different compounds. Just see, untreated and 2 hours we are increasing around 200 to 300 fold, but most of the cells did not decrease. But look at that, for example, it did decrease. So B2 somehow decreased the LPS induced. That means it is inhibiting the inflammation. Like that, we have done around 140 chemicals we have screened, and this is the second round we have to confirm. So even in the second round, as you can see, there are several chemical molecules we found they are trying to inhibit the inflammation. So which is mainly TNF alpha. We also checked the IL-6, which is another important cytokine. We found these molecules are capable of inhibiting the inflammation. This is the gene expression. And we also confirmed this by using ELISA, that is at the protein level in the supernatant. As you can see, naive followers, it increases around 20,000 folds TNF alpha protein, whereas upon drug plus LPS, it reduced that. This is what we are looking for. So these are chemicals which have very potential anti-inflammatory compounds. So we went back to the list because they are commercially available. We found several of them was already reported, which is actually good. We are identifying some chemicals which are already been reported. That means our system is working good. For example, let's say you have lung inflammation. For example, this is already well known. BET inhibitors are anti-inflammatory compounds and NF-kappa pathway inhibitors. So like that, we found several of the epigenetic inhibitors, some of them new, some of them already reported anti-inflammatory activity. Okay, so what we will do then next? We want to further test whether that's only in vitro what you've seen. Now we want to test model system. We will select some of them, and then what we do, we'll test in the mice model. We have to prove that yes, they can work against the arthritis or any other inflammatory diseases. For example, we are planning to do collagen-induced arthritis, where you inject collagen to the rats or mice. Then what happens? They will produce arthritis similar to the rheumatoid arthritis phenotype, and then we will give the doses of the chemical compounds. And then we see whether it is able to inhibit in the in vivo situation also. If they are in vivo also, then we have a very potential chemical which can be taken forward for the IND analysis and other human testing. Right. That's where I will stop. So just briefly, I'm sorry I might have taken more time. So please remember, I just told you inflammation is very important. Our body's defense system. It is working until it removes the harmful signal or the foreign stimuli and also begins healing process. It takes care of the damage and it takes care of the repair also. Okay, unlike the acute inflammation which comes and goes after removal, chronic inflammation persists for a long time and cause very detrimental effects. For example, rheumatoid arthritis, where all the bones are degraded because of the higher inflammation. It can turn into autoinflammatory, killing our own body cells. So the anti-inflammatory medicines include non-steroidal anti-inflammatory drugs, for example, aspirin, naproxen, ibuprofen. I told you COX-2 and COX-1 inhibitors, and also we have talked about steroidal drugs and also biologics, monoclonal antibody drugs. Right. So right now we want more drugs, novel drugs. Why? After some time, these drugs currently we are in use, the patient stops responding to the current medicine due to some molecular mechanism, drug resistance or the cells getting some mutations. We don't know, but they are not working anymore like they used to work. So doctors, they always keep changing based on their progress. So that means we need novel, safer anti-inflammatory drugs. Okay, for that, new research, modern research are needed, different methods. For example, I am working on the epigenetic way to find like that. If one can work on kinases, one can work on different enzymes, different pathways, right? So I hope you understand how I am trying to do using the epigenetic gene regulation to stop the inflammation. Okay, so with this, I would like to acknowledge a few people, Director, IICT, and Dr. Ramakrishna sir as chairman of our applied biology department, both of them for hosting me for doing my research at IICT. And I'd like to thank Dr. Sai Balaji Andugulapati, scientist. He's giving talk tomorrow. You just have to hear him. He's a very, very good scientist and he's my colleague and collaborator. I thank him for his support, and I thank Komal Walvekar. She's my PhD student who is doing most of this work. And also I thank Department of Biotechnology for giving the money and salary for doing this job. Okay, I thank you all, and once again I thank you all, organizing committee, principal ma'am, for giving me this opportunity. I'll be happy to take any questions you have.
All the students and faculty, if you have any doubts regarding about this talk, you can ask to the resource person. Otherwise, you can post it in the chat box also.
You can also contact me anytime if anybody has any doubt or want to discuss. I'm very happy to interact with the students or anybody. Okay.
Okay, sir. Really, it is great pleasure to listen to your lecture, sir. And really, it is very useful and informative for all the students and faculty members who participated in this webinar. Thank you very much, sir, for accepting our invitation and given opportunity to learn about mechanism and molecular techniques of inflammation. Thank you, sir.
Thank you. Thank you
Thank you.
once again. Thank you all for listening to me, and thank you all the staff and the college giving me opportunity. Okay. Pleasure to give the talk.
Now we can go to second talk, and I would like to invite resource person Dr. S. Tripathi Garu, Assistant Professor, Department of Pharmacology, Shiksha Anusandhan University, Bhubaneswar, Orissa. Hearty welcome you, sir, to this 2 days national webinar. And I request Dr. Mrs. B. Hemalata Garu, Associate Professor, Department of Pharmaceutics, to introduce today's resource person, Dr. S. Tripathi Garu.
Good morning, everyone. Myself, Hemalata, Associate Professor, Vijay Institute of Pharmaceutical Sciences for Women. It's my privilege to introduce the profile of Dr. S. Tripathi Garu. Dr. S. Tripathi Garu is currently working as Assistant Professor in SPS College, SOA University, Bhubaneswar from 2001 to Sir worked as Lecturer in Pharmacology in A.M. Reddy Memorial College of Pharmacy, Guntur from 2006 to 2007 and worked as Assistant Professor in Pharmacology in Vasavi Institute of Pharmaceutical Sciences under Andhra University from 2007 to 16 and also worked as Associate Professor in Pharmacology in Sri Vasavi Institute of Pharmaceutical Sciences from 2016 to 21. Sir has completed Bachelor's from Department of Pharmaceutical Sciences, Utkal University, Bhubaneswar, Odisha, and completed Master's from University Department of Pharmaceutical Sciences, Utkal University, Bhubaneswar, in the Department of Pharmacology. Sir has awarded PhD from University Department of Pharmaceutical Sciences, Utkal University, Bhubaneswar. Sir also has completed LLB from Deen Kanal Law College, Utkal University, Odisha. Sir has taught several subjects in his career, like advanced pharmacology, molecular pharmacology, clinical pharmacology, toxicology. Sir have attended several AICTE seminars, webinars, and has hosted many conferences. Sir guided around 32 students, pharmacology students, for their MPharm project works. Sir have published 30 articles in various national and international journals. Sir has received reviewer invitation from various journals like Asian Journal of Research in Biochemistry, Journal of Pharmaceutical Research, South Asian Journal of Research in Microbiology, Asian Journal of Biology, South Asian Journal of Research in Microbiology. It's our privilege to have you, Tripathi sir. We are very happy to have you. Thank you very much, sir.
Good morning, everybody. Am I audible?
Yes, sir. You're audible, sir.
Thank you for this elaborative description of me. Welcome. So we'll go for our topic.
Now we can share the PPT, sir.
Is it visible now?
Yeah, sir. It's now visible, sir.
Okay. So good morning, everybody. And before starting this, just I want to tell one thing to the students. Wherever you got any doubt, just write it somewhere. And otherwise you can send me a chat that, sir, okay, this slide we got some problem, this slide you can explain me of. Because whatever I'll be explaining, all those things will be based on the screenings. Okay, for psychopharmacological screenings, which may be new for many fellows now. Okay, so it will be totally one experimental basis. Those things I just will tell you on the theoretical approach, how to approach for these screenings. Okay. So my topic will be mostly based on what is this screening, what are these basic types of screening, then how we can go for screening of a drug on CNS, the general approach for the screening for drugs with CNS, then we'll go for nootropic screening, how to go for screening of drugs, those are working on the memory. So we can work it on the Alzheimer's disease, cognitive enhancers, nootropic, anything you can tell them. Those will be a burning field now. So we'll be seeing how to screen a drug in the nootropic screening. Okay, and I'll basically tell you without certain great infrastructure also, how we can go for screening, do screening on this topic or on the nootropics. Okay, so first, sir, the screen is totally visible to you or it is half or so, sir?
Yeah, sir. Visible, sir. But
okay, now, okay, is it okay?
sir, you can select the maximize option, sir.
No, I have just gone for
sliding option.
Now is it clear?
Yeah, sir. It's visible, but it's not in sliding option, sir.
Now I have kept it.
Yeah.
Okay. Okay. So let's quickly start. What is this screening? We are telling screening, we'll do this, we'll do that. First, what is the meaning of this screening? Screening means it is the word, it comes from through investigation. When we are going for finding out something through an investigation, the process is called as screening. Okay, so the main purpose of the screening is to determine whether the new substance are worthy for further attention to indicate which among them some relevant pharmacological properties. Suppose we got something and we want that, okay, this is having this effect. So we have to find out a scientific basis for that. So those process, that is called as only screening. So it is mostly done first to find out the pharmacological activity for what? For a new thing. Suppose you got some phytochemicals, some XYZ people are using it and you are telling that, okay, this can be used for this or we will use for this. So those things, or else some chemically unidentified substances. Like in previously inflammation, when sir was discussing, you heard that, okay, they have found out some chemical and those chemicals they are telling that they are telling you working as anti-inflammatory agent. Now, how will prove that, okay, it is working as anti-inflammatory agent? For that, you have to go for some in vivo model. You will take some animals, you will give them, then you will find out that, okay, it is decreasing this inflammation, these parameters of the inflammation, then you can identify it. Okay, so the first main aim of the screening is to measure the pharmacological activity of a new or chemical identified substance. Then next, it will see there are actually many ways of we can determine why we are doing, but basically these three topics, it encompasses everything. So second one is to investigate the function of endogenous mediators. Suppose we have read in hormones that adrenaline show these things, growth hormone show these things, these substance show these things. Suppose this COX is responsible for protecting action of GIT. So all these things, how could you know? Unless until we'll go for some animal models or screen them in some animal models, we cannot do it. Therefore, the second major thing why we are doing screening is just to investigate the function of the endogenous mediators. And another one is to measure the drug toxicity and unwanted effect. Okay, so we know that, okay, this drug has shown some effect. We found it out. Now, whether that drug is safe enough to take, otherwise imagine many substances are there which will be saving us from the microorganisms, which will be protecting us from microorganisms, which will be giving some XYZ effects. But can we use all of them? Never, because we know that every drug may be associated with some toxicity. So to find out whether that drug has got having toxicity or unwanted effect, we are going for this screening. Okay, so this finding out toxicity, again, there are different guidelines on it. Like we got OECD guideline, IPA guidelines, gradually we'll read them. We'll screen those drugs thinking that, okay, this drug show this much effect, so we can use it. This drug does not show this effect, so we can use it. Like that, we'll be finding out. Okay, then all the things, all the screenings based on how we are screening it, what we are screening, based on these two parameters, we can divide them into three parts. Okay, so first one is called as simple screening. What we are doing here, you use one or two simple tests just to find out the particular property. Suppose you want that the glucose concentration of the body decreases. Our drug decreases the glucose concentration. So in that, what we'll do, we'll just give glucose to the animal, we'll give our drug, and we'll find out the glucose level. So whenever we are doing this type of small procedures, those are called as simple screening. Okay, then next type is called as blind screening. Blind means we don't know the effect of that drug. Somebody has came and he has given us a substance and he told that, okay, this substance having this effect, can you find it out? Okay, we are predicting that this substance will be having this effect. That type of screening is called as blind screening. So there what happened, we used to detect the activity of a new drug which pharmacological activity is unknown, we don't know it. Okay, just this screening is done just to find out whether this drug has got some potency so that we can go for future experiment on this. Imagine those are called as your serendipitical discoveries, like digitalis. Okay, so they just, it is a chemical, they thought that, okay, it will be showing this effect, then they go for finding out the detail of that. Those type of things are called as blind screening. Then the next type is called as program screening, which all of us we are following now. This means we know that this drug has got a specific type of effect, or it is showing effect on specific system or specific path. So we are going for screening that activity only. That is called as program screening. We know it is showing effect on this. So we'll go for screening on that part only. Suppose we know that our compound is increasing the memory. So we'll go for screening the drug on different models. Those are available for memory screening, nootropic screening. Okay, so that is called as a program screening. Here what we do, we go for quantitative assay of the compound and comparison. Two things we involve, quantitative assay, comparison. How much compound or how much drug substance is responsible for showing the effect. That is called quantitative. And we go for comparison. Suppose we have taken a drug, it is working as an anti-inflammatory agent. So there are already in the market, there are a lot of anti-inflammatory agents. Now what we'll do, we'll compare our drug with that and we'll tell that, okay, this drug, our new drug, decrease the inflammation as compared to this drug. Therefore, this can be used as an anti-inflammatory agent. So those are called as the comparison with a standard drug. These are the basic three types of screening. Okay, one is called as simple, then it is blind, then it is called as program screening. Then we'll go for directly, let's come for our topic, nootropic screening or general procedure for screening of CNS drug. Okay, so this general procedure of screening of drugs, has been first proposed by Irwin in 1968 in mice. Okay, so that test is called as Irwin's test. So in respect of him, we are keeping this test as Irwin test. This is one of the most accepted model for screening of any type of CNS drug, any drug working on CNS, that we are going for Irwin's test. Okay, what we are doing here, this is a observational screening paradigm. Paradigm means different parts or different types. Paradigm, that comparison of a battery of tests used to assess the mouse or rats neurobiological and physiological state. Parameters evaluated include autonomic, sensomotor, convulsometer. All these things we'll discuss in the further slides. So this is just an observational screening. Different parameters just in the animals we'll observe. We'll give the drug. We need not to induce anything. Just we have given our test compound and we'll observe what the animal is showing, what behavior it is showing. Okay, so he has proposed a group of behavior. Actually, there are 4 categories of behavior we'll discuss. So when we are going for screening of those 4 categories of behaviors, that is called as Irwin's test. But as he has told in the mice, when you are doing the test in the rat, that is called as modified Irwin test. Remember it. Irwin test is on mice, whereas modified Irwin test is on the rats. Then, based on these studies, our safety pharmacological studies developed some parts of this. So that is called as functional observation battery test. FOB, functional observational battery test. When we will read further in the toxicological things, toxicity, that time we will come across this. Means when this Irwin test, whatever he has told, we just modified some parameters of those tests, some evaluation parameter, some testing parameter, that became the functional observational battery test. This test is mostly used for different types of drugs whenever we thinking of safety pharmacology. Imagine one drug has, you have proposed a drug. Now this drug is showing this effect. Now I have to go for toxicity study of that drug. Okay, so I have found out the LD50, I found out the long-term toxicity. Now we have to see that on which organs, how this drug show effect. So that is called as functional observational battery test. Okay, so we'll generally discuss these 2 procedures, how they are and how to find out. And remember, for doing all this work, we don't require some sophisticated instruments. I'll show you how we can do it easily in a lab without much expenditure or so. Okay, so this is you can call as functional observational battery, and this is your Irwin test. So as I told, there are 4 parameters we are observing. We are observing autonomic parameters. We are observing neuromuscular parameters. We are observing sensomotor parameters, and we are observing behavioral parameters. Okay, so this line, it indicates this is accepted for functional observation battery and this is accepted for Irwin's test. Okay, so for the timing, you can understand, imagine that everything we can study, everything we have to study. Okay, so in autonomic parameter, what we'll observe? We'll observe pupil reflex, we'll observe pupil diameter. Reflex means whenever somebody is approaching towards your eye, automatically your pupil get constricted. Okay, so that is called as pupil reflex. Pupil diameter, and the same thing in FBP, we are telling it is as pupil reflex. We'll observe salivation, we'll observe lacrimation. Salivation means saliva. Pavlov experiments, food is nearby, automatically saliva secretion will be there. So that is your autonomic. Lacrimation, if pain is there, you cannot express. It is just tears will come out. Piloerection. Piloerection, everybody knows your skin hairs will erect up. Then excessive urination, that we can measure by metabolic age. Diarrhea, loose fecal materials. Abdominal respiration, when you are sleeping, imagine your abdomen respiration starts up. Whenever you are working more, your thoracic respiration used to be there, that one. And rectal temperature. These things are observed as autonomic responses. Okay, then in neuromuscular things, we'll observe the abnormal posture, how the animals pose, that we'll observe. We'll observe the abnormal gait, how the animal's presentation is there. We tell a get up, means your gait, how your positions are there. These things I'll show in the pictures, though some videos are there, it is difficult for me to show it up. I'll show you these things in pictures. Then catalepsy. Catalepsy, those who have read pharmacology, you know that haloperidol and all antipsychotic drugs induce catalepsy. Catalepsy means it will be something like paralyzing one part, means you cannot move that part. That is catalepsy. Body tone, your body, how it is there, whether it is bending down or whether it is curved up, those things. Grip strength. Grip means this one. Imagine whenever you are sedative, you cannot hold something properly. So that is called as the grip strength. We will see the tremor in that. Shiverings. Shiverings means everybody knows it. Twitches, convulsions, these all are excitatory things. Whenever epilepsy occurs, when imagine a CNS stimulants you have given. Okay, so that cases it will go for twitching, convulsions, everything. Starved tail, this is one particular thing. Whenever the animal lift up its tail and it used to move, that is called a starved tail. Ptosis, exophthalmos, all these things will observe in the neuromuscular thing. Okay, then in sensomotor, we will see the approach response, means suppose the animal is here, some fears come, so automatically it will show some response. Imagine now this animal has taken a sedative drug. Now it will not respond at all. So that we'll see. We'll see the touch response. Just have a touch on the animal, it will show some response. That we'll see. Subtle response. Subtle response means imagine whenever suddenly you heard a huge sound, large sound, or something unexpected thing, you show some response, like your piloerection will be there. You cannot decide what to do. That is subtle response. Then palpebral reflex, your eye opening, that one. Pinna reflex, you know that this mice or rabbit, they got some pinna, just if you touch, they used to move their ear in certain way. That is the pinna reflex. Tail flick reflex, some pain is there, automatically withdraw, that is tail flick reflex. Writing reflex, that I'll explain in the diagram. Writing means write. Suppose you throw some animals at the top. Do you think it will fall on its back? It used to rectify. It used to go for somersault in the air and it used to rectify itself. That is called as writing reflex, or that is called as landing foot spiral, also same thing. Then grasping reflex, traction reflex, how much it is gripping up, that is grasping. And traction, how much their body, how much it can resist, how much it can go for expanding, that is called as traction reflex. These are all things we can go for sensomotor. Then certain behavioral things like arousal or sedation, whether the animal is active, whether it is doing its work or not. It is how it is handling to the reactivity. It is highly reactive or not reactive. It is aggressive or not aggressive. Whether it is doing its spontaneous activity or not. Whether it is rearing. Rearing means it will just poke up its nose and it will see outside. That is rearing. Sniffing, grooming, scratching, all these behaviors we can see. All this you can in your lab also you can see. Just take an animal, keep it in a big beaker. Take a small mice, keep it in a 500 to 1000 ml beaker. Now you go on noting how much time it is standing up and it is touching the walls. That is one we can observe. You can observe how much time it is scratching itself. That is its. Okay, sniffing, automatically the animal in its nose, it will go for sniffing of that. It will smell it. Okay, so its mustaches will be moving up. That you can observe. You can easily see, take a normal animal, see the effect. Take an animal by giving him chloropromazine or any sedative drug, see the effect. Easily you can know the variation between these two. Stereotype. Stereotype effect means when continuously repeated movement, illogic repeated movement animal is doing, that is called as stereotype. Okay, like imagine some fellows automatically in the class also, they used to move their legs without any logic. Some used to move the pens in certain way. Some used to touch their hairs like this. Okay, so all these things are called as stereotype behavior. Vocalization, it will make sound. Normally animal does not make sound. Sometimes it used to, whenever it is pain, they will make sound. So we'll observe all these parameters and based on that observation, we'll give them some scores. Like it is showing, we'll give 0. It is not showing, we'll give 1. Okay, so based on this, we'll give them different scoring. So now all these things, whatever we'll get, I'll show in a picture and we'll go for explaining. So this is suppose our gait. This animal you see, what it is doing? Its legs are slightly scratched up. And this animal you see, the leg is wide apart. Means the drug, whatever it has given, it has shown some effect on the neuromuscular junction or on the CNS, which does not allow this animal to get his paw back. Okay, so this is the abnormal gait. The animal could not stand properly. See this, both the legs are flaccid off. See this, when this hand, this foreleg is flaccid off. Okay, so this is normal animal. Here, whenever we are giving the drugs and whenever the drugs has been given, the animal's gait has been changed. Is it, you can observe, see this leg, how it is. Both the legs are gone for wide apart. The normal animal, legs are compact. Now the legs are gone wide apart. Means this drug is affecting the gait. So based on this, we can give them some score. We can give it 1, 2, 3. We can give the scores on that. Okay. Next, this is an example of a posture. Normal animal looking towards you, nothing. Now this, see this animal, what happened? This animal is be drowsy, it is moving away. This animal, see, it is totally looking down. Okay, so this is the abnormal posture of this animal. This is on one side, this one, you see, this is in another angle. Okay, this is your abnormal posture. Posture of the animal is not correct on this. Okay, then this is a part of grip strength. Grip strength means how much animal is gripping. You can, this is just a sensiometer where you are seeing how much it can drag up. You can do this experiment in your lab by taking this rotor rod apparatus, or else you just hang a thin wire and keep your animal on that. Okay, so that it will be holding up and staying. Just you can see how much time it is staying up. That way you can do your grip strength test. Then this is turf tail. See, this is our normal animal. Now this animal, it has heard something or some noise or something. So what happened? It has just erected its back. Okay, this type of observation is called as turf tail. Okay, then this is catalepsy. This is a classic model for haloperidol induced catalepsy. In this, what we do? We keep the animal in an awkward position. See, normally 4 paws should be on the ground. Now imagine if one paw you kept it here, the animal tends to bring back this paw. It should bring back its paw, but if cannot bring back its paw, means this drug has got cataleptic property. It does not allow the animal to rectify itself from an awkward position. Okay, so this is hind limb catalepsy, this is forelimb catalepsy, this is back leg, and this is a forelimb. This animal is supposed to drag it up, but it cannot drag it up. This is called catalepsy. This is easy model you can do by taking haloperidol. Take haloperidol drug, give it to the rat or mice. It will go for catalepsy. Then you can keep the animal in an awkward position. The animal does not rectify it. If your drug rectify that one, you can tell that, okay, my drug is anti-cataleptic drug. Okay, so this is called a subtle response. Suddenly hearing some abnormal sound or something, this is called a subtle response. Okay, then people got doubt on this ptosis and exophthalmos. That's why this picture, which contain both the things, I kept it. See, ptosis means dropping of the eyelid. Eyelid, this animal will just drop its eyelid. It will close its eyelid. Okay, that is called as ptosis. Whereas exophthalmos means bulging of eyeball. This eyeball used to protrude out. It will come out. Protrusion of the eyeball, or you can call as bulging of eyeballs. Okay. Then one classic model is there. We call it writing reflex. Writing means it makes itself right. It rectifies itself. So in this case, what we are doing? We are holding an animal from the tail like this, and just we are flicking it up. Okay, opposite direction. So what happens when the animal moves up? It rectifies itself and it present on the ground. It comes to the ground like this. If the animal rectify itself and come to the ground, that indicates that this animal have not lost the writing, and the drug does not have any effect on the writing. So we are giving it a score 0. Okay. Then what happened? When it falls down, normally it makes it tail. When the mouse, imagine, it will be weak and flaccid tail, we can give them score 1 or 1.5 that. Or it has got gait problem. Means after falling down, the animal should walk up. It should walk. So if it does not walk and the legs have been flaccid up, we can give them a score 1. Okay. Then if the animal is walking, but it has got unsteady gait, it has got ataxia, okay, or it is showing so reflexes, that case it will give them the score 2. Okay, if the animal has got complete limb paralysis, we are giving them the score 3. Okay, so when the animal has got all the 4 legs paralysis, that we call give them 4. Or if it does not move, the way you throwing it up, it has fallen like that, that indicates that it is moribund animal, or else the drug has got complete sedation effect on it, or the animal has been died out. Okay, so based on writing reflex, we are giving these scores, and based on that, we are measuring how our drug works on the animal. Okay, then these are some behavioral assessment what we'll be observing in the animal. What this is the climbing behavior. As I told you, imagine keep a beaker here, so the animal will look towards the up. That is called as climbing. Animal is walking. Rearing means 2 legs in the down, then 4 legs it will up, and it will move its head like this here and there. Okay, so that is called as rearing. Sniffing in its nose, it will go for sniff up. It will just smell them. That is called as sniffing response. Then these are called as licking, forepaw licking. It will this is forepaw licking, this is hindpaw licking. It will just lick up the forepaw, front 2 legs, back 2 legs. This is called as body grooving. Means if some responses or something is there, what happen? Animal used to move up. It will move. Suppose the place is not there, but it will move up like this and it will come back. Okay, so this is called as body grooming, and this parameter, this is called as face grooming. Okay, on its face, it will, in its 4 legs, it will just clean up its face. Okay, so that is called as face grooming. Okay, this is called as focus exploration. When we are seeing the explorative behavior of the animal, this is called as head dip, this is called as edge sniff. This is a model, it is called as hole board apparatus. See, these are 2 holes. Suppose you can design it of yourself. Okay, so take a board and make small holes on it and cover up that. Leave the animal over there. So what the animal will do? Animal will sniff the edge first, then it will drop its head. Okay, so this will be head dip. Okay, and immobility means animal does not move at all. Okay, so these are the basic screenings what we are doing as a blind screening, or this is called as the functional observation battery. Whenever some drugs, you know that okay, it may show some toxicity, you can observe all these parameters. Based on these parameters, you can tell that whether your drug has got some toxicity or not. Okay, so that's why we call it functional observation battery test. Imagine, remember that this will be mostly used for safety pharmacological assays. Okay, so then we'll go for this nootropic. So, and remember, from all this, we'll be getting a basic idea that on which system or on what drug will work. How you can go for the next screening. Suppose you are designing next what to do, whether we'll go for anxiolytic agent, whether we'll go for antipsychotic agent, whether we'll go for antidepressant effect, whether we'll go for any other type of psychotropic substances or neurological effect. These are the basic screening based on which we can go for designing our next experiments. Therefore, it is called as Ewing's blind screening. In safety pharmacology, we are telling it functional observation battery test. Okay, we'll directly next we go for, these are gone for general screening. Then we'll go for nootropic screening, my own topic, how to screen of nootropics, what are these nootropics, why that is that much important in current scenario. Okay, so this nootropics means, nu means mind. Okay, in Greek, this nous means mind, tropes means direction or grown up, you can tell. Okay, so this nootropics comes from a word. Nootropics means the drugs, those who give a direction to the mind, or those who have got some effect or some protective effects on the mind or on the thinkings, right? So it is these are the drugs, those are giving some effect on mind. So it is called as, otherwise smart, we are telling them the smart drug. Some people think that if we eat nootropics, we'll remember everything and our exam will be good. That does not work practically. It takes time. Okay, slowly you have to modify. There are a lot of things on this. Okay, so we can call them our cognitive enhancers. Okay, because what they do, they enhance their memory and cognitive function. Cognitive means holding up, remembering the things function. Okay, so it is proposed by the Belgian scientists K. Gijura and V. Scondia. These two scientists actually proposed the name nootropics. Okay, so what these nootropics do, how they increase the memory. Actually, up to now, we don't have a specific mechanism that, yes, this is the only thing which by which increase memory. There are a lot of theories on it. We'll read, these are the some basic theories based on which it may show effect. So first, what it is proposed, the impact on the learning and memory. But how they do? They impact higher cortical functions and mental retardation. Whenever you, what happened? Memory means whatever you got, how your mind interpret with that. Okay, so it involves your whole brain. Okay, so if anything which has altered the cortical function, cerebral cortex functions, or gone for the mental retardation, so they do for modifying those functions. They have got psychostimulating property. Why we are telling psychostimulating property? Because they have got influence on the intellectual motor retardation. Suppose you tell, I don't want to do anything. Why, I don't know, but I am not in a mood to do anything. That is called as intellectual or motor retardation. I don't want to go. I not feel like to do these things. So that is called as apathy or mental inertia. You will not do those things. That is called. So in those cases also they will be working. They have got anti-aesthetic property, asthenia, lethargy, weakness. So these drugs, what they do, they stimulate your brain. Okay, they have got adaptogenic property, means they increase tolerance for the extreme exogenous factors. Like suppose exogenous toxins are there, some free radicals are there. So these substances are there, they will be protecting yourself from there. They have got anxiolytic effect. Okay, this is just a theory. They decreases the anxiety. Now, what is anxiety? Imagine repeated thinking on the same thing. Okay, what is anxiety? So how they work on that? They work on liability, irritability, anxiety. Why you feel anxiety? Imagine, because you are thinking on a particular thing more, much more. That's why. Okay, same book you are reading, you will not feel anxiety in normal days. During exam time, if you read, you will feel anxiety. Because of, why? Because you are thinking that I have to do good in the exam, that I have not read, what to do. Okay, so imagine in your brain at the back, these questions are going on. So if you are decreasing those questions, automatically you can get rid of that anxiety. So these drugs, as they increases your cortical blood flow, as they integrate your brain functions, they have got this anxiolytic property. They got neuroprotective property, because what they do, they go for modifying the cerebral circulations, they go for increasing blood flow to that area, different parts of the brain. So they do that. They facilitate intra-hemispheric transmission to the CNS. Your right hemisphere, left hemisphere, parts of hemispheres are the frontal, parietal, all these parts. So whatever this transmission is there, they used to facilitate all those things. So as they facilitate, we are telling them they are the smarter. They make the people smart. You can think of more things on that. You will not feel stress, anxiety. You can remember more things. So that's why there is a raw name point for it, that these drugs are called as the smart drugs. Okay. So all these functionality, how they do. Okay, as I told, specific mechanism up to now yet to be discovered. We have discovered only one neurotransmitter, acetylcholine. Then another one is discovered, NMDA, decreasing expression of NMDA, increasing expression of acetylcholine. Other than that, they have got these different mechanisms. They increases blood supply of glucose and oxygen. So as your body will be getting more oxygen, more glucose, so automatically the process will be faster. The things will be produced quickly and the work will be normal. Okay, it has got anti-hypoxic effect as it provides oxygen. It has got anti-hypoxic effect. It protects brain tissue from neurotoxicity. What happens? It protects the brain from different free radicals, different heavy metals, different other enzymes. It protects it. Then they have got positive effect on neuronal proteins and nucleic acid synthesis. Brain, whatever neurochemicals are synthesized, they used to stimulate the positive effect on that. They stimulate phospholipid mechanisms, neurohumoral transmission, everything they go for stimulating it. They affect the elimination of oxygen free radicals in your brain, liver, all these parts. Every time work is going on, so automatically there are some free radicals are forming up. So those free radicals go for cell damage or tissue damage. So they mostly eliminate that and they improve blood flow in the brain. Okay, so these are the basic way how the nootropics are working. Okay. Then we'll come for the screening methods. Now, okay, we know that, okay, this is a compound. This is a nootropic compound. I have got a nootropic compound. Suppose XYZ plant, it is used as a nootropic compound and I have found it out. So now I just want to go for screen. I want to give a confirmation result that my compound is having a nootropic effect. Okay, so for that, we are using two types of methods. Okay, either it is in vitro methods or in vivo methods. In vitro means when you are doing outside the body, when we are not using the whole animal directly, that is called as in vitro, outside. In vivo means in the body. Whenever we are doing that experiment on a body, on a living animal, we are doing, that is called as in vivo method. Okay, so in vivo memory, we got three basic type of screening.
Sir, excuse me, sir.
Yes.
Sir, same slide is visible, sir.
Which slide is visible, sir?
Screening of drugs, sir. Screening through investigation of drugs, sir.
Oh, why?
Maybe.
Okay. Now is it visible? No.
No, sir.
Please change the slides.
No, I have
Please change
already
the slide,
changed.
sir. Change the slide, sir.
1 minute, I'll go for again sharing it.
Yeah, sir.
Now is it visible?
Yeah, sir. Now it's visible, sir.
Okay, right. Okay, now it is there or from the earlier it was there?
Now it was there, sir. No problem, sir. Now it's visible, sir.
No, other slides were not also not visible?
Yeah, sir.
Then you should have told me, sir, earlier, because from my side it is visible. Therefore, I am going on telling it. Then should I repeat those or I'll show those pictures again?
Just you move the slide, sir.
See,
You can understand the concept, sir.
is it visible now?
Yeah, sir. Now it's visible, sir.
You should have told me because these pictures, this. Should I go for explaining all these things again?
If it is possible, it's very much helpful for us, sir.
Then you should have told me, sir, because I thought that it is slides are moving, so I am going. Okay, this slide was visible earlier, or it was also not visible?
Yeah, sir. It's now it's visible, sir.
Then earlier, okay. Okay, this not to here now. This rat slide is visible.
Yeah, sir. Visible, sir.
You should have told me, sir, earlier it is not visible. I could not. I have not seen it practically. Okay, not a problem. Go for. Okay. See, okay. There might be some technical issues. I don't know if something is problem. Just anybody can unmute and you can tell me. Otherwise, you can tell the concerned faculty so that they'll tell me off. I was thinking that if everything was visible, therefore I'm going on. Okay, see kids, this is abnormal gait. See this animal, these two legs are okay. This animal is moving forward. Okay, so this gait is, you can tell that this animal is presenting properly. Gait is okay. Now observe this animal. See this animal. What happened? The legs has been expanded up. Means he cannot move properly with these legs. This leg. Because why? It may happen that the drug is having a neuromuscular problem, or the drug has affected something in the CNS, or it is depressing CNS, therefore it cannot drag it up. Got it? So here you see both the legs, they have been expanded up. Now this animal, you see both the legs have gone flaccid and outside. So this is abnormal gait. Whenever you are making the animal to place on a surface, the animal could not stand properly or cannot move properly. So that when we are observing, this is called as abnormal gait. Right? This is abnormal posture. See this animal looking at towards directly, and this animal, you see his eyes and see its orientation. What happened? It has gone bit down. This animal has totally dropped it and become abnormally down. So this is abnormal posture. This is from one angle. This is from a mice another angle. You see, See this animal, head is erect, now head is down. This animal, head is totally down. See its ears, ear has to be this like this. Now the ear has also dropped up to the side. Okay, so these are called as abnormal posture. Okay, then this is grip strength. Grip strength, this is a meter. We'll just keep the animal and we'll drag it up. We'll see how much it is dragging it. You can do it if it is not available also. You can work it on the rotor rod apparatus. You can see it, or else on a simple, take a thread, keep two holders, tie a thread in between that, a thick thread. Okay, thread or a thin pipe. Just make your animal to claw it and stay there. You see how much time it is staying. If it is falling down, grip strength is less. Okay, this is turf tail. Animal will go for deflecting its tail. Okay, this is called as turf tail test. This is called catalepsy. Animal could not rectify itself from a wrong position. This is called as catalepsy. This is called as subtle response. Means when you are observing some huge sound or something, so what happened? Animal go for some abnormal response. It used to move here and there, lifting up. That is called as subtle response. Okay, this is ptosis and this is exophthalmos. Ptosis means it will drop its eyelid. That is called ptosis. Exophthalmos means it will protrude its eyeball. See, its eyeball has protruded out. Otherwise, it has came out. So that is called as exophthalmos. Okay, this is writing reflex. As we explain, what the animal will flick it up and we'll see if the animal healthy mouse, what happened? It will fall down like this. If the tail, it is falling down and the legs are flaccid, animal could not walk much sufficiently, that is called as, there will give the score 1. If the animal is cannot move properly, ataxia, will give score 2. If the complete limb paralysis are there, back limbs, then we'll give score 3. If it is 4 limbs paralysis, we'll give them score 4. And if the animal does not move at all, the way it has fallen, suppose it has fallen like this back, it could not rectify itself, we'll give the score 5, or else you can tell that this is a CNS depressant drug or the animal has died off. Okay, these are the basic behavioral assessment of the animal. This is the climbing behavior. Animal stand on its 2 legs, giving a force on the tail, lift up with this leg, and his 4 limbs will be towards the front. That is called as climbing behavior. Walking normally, you know, it will walk properly. This is immobile sniffing. Sniffing means animal will stay down and it will sniff. Okay, so this sniffing can happen in the climbing behavior also. If you imagine keeping a beaker here, this animal will sniff to the surface of the beaker now. So this is called as a sniffing and rearing also can. This is rearing. The animal will see, observe. See these 2 positions, climbing position and rearing position. Climbing on this leg and in the tip of the tail, this body part is not on the ground, right? Here you see that this body part is on the ground. It is rearing. It has kept its lower body part totally on the surface and it is observing. That is called as rearing. Here it is straight enough. So that is called as climbing. Okay, then this is the licking behavior. This is front paw licking. This is hind paw licking. It will just on its tongue, it will clean up its paw. We can see it in this what is that analgesiometer, you can observe it up. Okay, then this is called as grooming. This is whole body, it will move, or it is only face, it will move its face and it will do something, again it will come back, that is face grooming. This is focused exploration. This is seen in the anxiolytic or sometime in the memory also, you are observing it. Here this is a hole. We'll do it in the whole board test. This is a hole. What happened when the fast the age has came, the animal will think that I may fall down. So what it will do? It will sniff it up. Okay, when it confirm that, okay, through this I will not fall, it will poke its head down. So this is sniff, edge sniff, and this is head drop. And this is immobility. Animal will not move at all. Okay, then these things are enough to explain it again, I think, sir. Okay, is it visible now?
Yes, sir.
Okay, fine. So, yes, is it visible, these slides?
Yes, sir. Visible, sir.
Okay, fine. We'll go for this in vitro methods. In vitro means we are doing the things outside. We have taken some enzymes, some proteins, and outside the body in the lab setup we are doing. We can see in vitro inhibition of acetylcholinesterase activity, because up to now whatever drugs has been discovered, they have shown that their effect on the acetylcholinesterase, like your galantamine, rivastigmine, all those drugs, they work on acetylcholinesterase. So if we are doing taking acetylcholinesterase and we are seeing our drug is inhibiting acetylcholinesterase activity, that indicates that our drug inside increases the acetylcholine concentration in the brain, so it can work as a nootropic. So we can see that, we can see in vitro butyrylcholinesterase activity, we can see ex vivo cholinesterase activity, we can see the binding release of acetylcholine from the rat brain slice. Take a brain slice, normal brain slice, and you give your drug. If your drug is releasing acetylcholine, that you can measure by radioimmunoassay, that we can see. We can see oxytryptamine binding muscarinic receptor in the forebrain. We can see the stimulation of phosphatidyl inositol turnover in the brain. Okay, this is the secondary messenger for acetylcholine receptor. We can see the effect on the cultural neoglia, astroglial cells. We know that, okay, if your in Alzheimer's disease, what happened? Brain used to get degenerated. Suppose your drug decreases the degeneration or increases the proliferation of these cells, that indicates that they are proliferating in the brain, these cells, so they can work as a nootropic agent. So in vitro method, we can go for all this, and there are a lot of methods practically. Just for the example, I kept some methods. Okay, then we'll go for in vivo method. In vivo, as we discussed, there are basically 3 types. Okay, inhibitory avoidance or passive avoidance, active avoidance, and discrimination test. Okay, discrimination. So first we'll see what is this passive avoidance. Passive avoidance means automatically you are avoiding something. Suppose fire is there, you will not go to the fire. Why? Because you already know that if I go to the fire, something will burn up or I'll get a pain, so you are avoiding it. So these type of things are called as passive avoidance. You learn already that, okay, we should not go there, and you are not going. Suppose you are going there, that indicates that you are not in a sense, or your memory has been lost. Suppose somebody doesn't have remember, somebody has gone for loss of his memory. Now he can walk anywhere, he can do anything. So that work, that type of screening whenever doing in the animals, that is called as inhibitory, or that is called as passive avoidance test. Okay, but it is immediate activity learn it where inhibition to the immediate activities or learned activities. Whatever activities already been learned by the animal, that it will not do, it will inhibit that one. Okay, so it is employed to describe experiment in which the animal learn to avoid a noxious event by suppressing a particular behavior. Okay, it will be not do one activity, not do the intended activity what it is supposed to do. Okay, so in this first one is called as step down method. Step down method means, I'll show you in that diagram side. So see, this is the step down method. Okay, step down method what? Animal normally, if you kept an animal here, what it will do? It tends to come down. Okay, now what you do? You keep a sock here. Okay, you keep a sock here. Whenever the animal is getting a sock, do you think it will come down? It will not come, it will stay there. So you observe for 5-6 minutes. Just give a sock, let the animal go up, and you observe that animal for 3, 4, 5 minutes based on your protocol you observe. Now, if the animal does not come down, that indicates that the animal remember that, okay, if I'll go down, I'll get a shock, it will not go down. So these type of things are called as step down. Okay, step down method. Okay, next method is called as step through. Step through is this one. Okay, this is called as step through method. Step through, what happened? You keep the make a chamber, one is dark, one is light. Normally the animal's behavior that it will come to the dark part. It will come to the dark part. Okay, so whenever you are keeping an animal here, the animal used to come to the dark. Normally it will come back. Okay, so here now you keep a sock or keep a sound so that the animal will reflexively go back to this place. Imagine the animal don't want to come here, but as it remember that there is some modalities here which may cause harm to him, it is not coming. means it is remembering that, okay, something is there. So these type of things are called as step through. Okay, this step through, you can do it in two ways. Step through, another modified version is called as two compartment test. This is your two compartment test. Step through, you can make a chamber, you can keep different beams. Suppose the animal is walking, you keep that, okay, here shock will get. So the animal walk in this fast, very fast. Step through is a old method which has been modified as compartment, two compartment model. Okay, then uphill avoidance. Uphill avoidance is same as your step down or step up. Okay, uphill avoidance means this animal will not come down again. That is called as uphill avoidance. Okay, because all the animals are negative geotaxis, means they will be towards the top. Okay, so that will be called as uphill avoidance. This you can do it in another way. You can keep a slant base like this. Okay, slant base, a 30-degree base you can keep. Leave your animal here. So automatically animal wants to come up. Okay, you keep some food here, the animal will tends to come up. Now you keep a shock in between, or keep something, if the animal come, it is getting a brick shock, or you make some unusual sound. So if the animal is coming towards middle, you make that sound. So animal will see that, okay, I'm getting sound here, so it will not come. If the animal still then coming, that indicates that it does not remember, right? So you give a drug which is allowing the animal to lose its memory, like your scopolamine, or you can go for a degenerative drug, galactose or everything, anything you can give, which will go for degeneration of the brain, or it will go for loss of memory. Now you screen your drug. So in this scopolamine induced case, what happened? The animal will forget. So it will be going. Though it will get the shock, it will go. Okay. So these methods are called as passive avoidance. Means animal has got its inner tendency to avoid something, it is not avoiding. That is passive avoidance. Then it is called as active avoidance. Another method is called as active avoidance. This is bit higher center as compared to passive. Active avoidance means you train the animal that if we will do this, this thing will happen. Okay, if there is a sound, like your Pavlov experiment when we read in +2 or so, Pavlov experiment is there saliva, that it is a modified version of that one. Okay, so in this case what happened, you keep two compartments, okay, or two areas, the one area and a whole another area, okay, gap another area. So you give a sound, okay, just after giving the sound you give a shock. So whenever you are giving a shock, the animal is coming to this side, right? So now whenever you make the animal to habituate on this, give a sound, give a shock, animal will come here. Give a sound, give a shock, animal will come here. Now whenever you are giving a sound, animal remember that I will get a shock now. So whenever you are giving a sound, the animal will come this side. So animal learn that whenever sound will be there, just after that there will be a shock and I have to avoid that. So this type of avoidance test is called as active avoidance. Okay, this type of things when we are screening, this is called as active avoidance test. Okay, so this can be done in various ways. This is just a one example I have shown you. We can think of, or there are various methods of active avoidance. Okay, that we can call as runway avoidance, we can call as jumping avoidance, anything, everything will be similar to that. Yes, roll number 50, 46 or 50. Ask. You raise your hand. Okay, if you want to ask something, ask roll number 46 or 50. 217N1R0046 and 50.
Yes, sir. It's student's roll number.
Yes, he has raised his hand. Or she has raised her hand. Do they want to ask anything? Just sir, ask them whether if they want to ask something, they can ask me, not a problem. Okay, then we'll go for discrimination learning. Okay, these two gone, the avoidance. Now in memory higher version is called discrimination learning. Means you should able to discriminate between some modalities, some things, some different, some similar things you have to discriminate. Okay, so in this we call partial discrimination. Means if there are two things similar to us, we have to discriminate that, okay, this part we have to go, this part we should not go. So in this case, what happened? Suppose this is our noble arm. Here we keep some food and train the animal to go this side. Okay, leave the animal here. This is called this discrimination by Y image. Okay, here you leave the animal this part and let this train this animal to go to this side as food. So this is your right entry. Therefore, we are telling it as a noble arm. Okay, so if the animal is going here, means the animal are memorized that, okay, I have to go this side. Okay, you train the animal like this, then you open up all these arms. So imagine if the animal has trained properly, remember, then it will go to this arm. If the animal does not remember that, it can go to this arm, all these areas it will move. Okay, so this way we can go for discrimination. This is Y image. Okay, so these things you can take it in a camera in your mobile phone, you can just record it. Later you can analyze that. Suppose in 5 minutes, how much time animal is going this side and this side. So if it is going more time this side, then your drug is having nootropic effect. If it is going both the sides similar way, then this drug does not have any effect on the memory. Okay, so this discrimination can be seen by various ways. This is one Y image. This is same thing like that. This is called as partial discrimination by radial image. Here we observe two similar things. Here we will observe in 6 or 8. This is a 8-stage model. We'll see. First, you train your animal to go to certain part. Suppose these 4 areas, I'll train my animal to go. Okay, so I leave this animal here. So this animal will go to the, I'll train him, I'll close all these doors and I'll train him to go to this part, this part, this part, or this part. Okay, after training, now just leave the animal after training, after giving your test substances. Now, if the animal remember the things properly, it will go to this area. Otherwise, it may go to this area. So if it is going to this area, that is correct. You can give him score 1. If it is going to this area, you give him a score 0. Now you observe how much time animal is going to the open area and how much time the animal is going to the targeted area, how much time it is going to the non-training area. So based on that score, you can find out whether a drug is showing anti-emetic nootropic effect or not. Okay, this is another classical method we call spatial learning by water image, which is proposed by Morris in 1984. In this, what happened? We'll take a bucket of water. Not bucket, it should be actually 1 meter. You can take a variation. Okay, the diameter should be 1 meter. In this, you will keep a platform, right? We'll keep a platform and water here. Okay, so if you are leaving the animal in the water, what? It will not stay here, so it will swim. So now you will leave the animal, suppose here, this is my part, I leave the animal here, and this animal, what it will do? It will go, it will swim to this area. Okay, so train the animal to go to this area. Do you think it will obey your rule and it will go like this? You have to train him that, okay, this is the shortest way, so you have to go. You have to go in this way. So you see, he has given different identification. That animal has to go in this way or animal has to go in this way. This will be the shortest area. Okay, it can go in this way, but we cannot train the animal to go this way. Therefore, here the things we have given. Okay, now you just go for training the animal. So this animal automatically trained to go to that part. Once this animal is trained, now you give the drug which is causing amnesia, which is means causing CNS damage or amnesia, and give your test compounds. Okay, so as you have trained your animal to go here, so without seeing, it can go to this place. Okay, so after that, in test part, what you do, this your water container, you add milk or add some white substance which will be not irritating. Normally milk we are adding because it will not irritating, and just above that platform keep the label. Okay, so this animal could not see this platform now, could not see the hidden platform. Okay, because it is all over white, it cannot see this platform. Okay, but earlier when you have trained, animal knows that if I will be left in this place, I have to go here. Now it's supposed to go this part without seeing also. It can discriminate and it can go. If the animal goes there in, you have to observe how much time it is going. If the animal going in a less time, That indicates that drug is working as a nootropic drug. The animal can remember that I have to go there and it is going. If it is going through some other parts or the time is much more, then based on that you can tell that your animal does not remember or your drug doesn't have efficacy. You can just observe the time, you can observe the pathways, how it is moving. Okay, based on that you can find out the result. Okay. So these things, this discrimination, the spatial discrimination can be done by various measures. These whatever 3 measures I told you, those are normal, generally used in all lab setups. You can easily make it. Other than that, there are various ways. You can go for a T image. Y image it is there. We can make same like that T image. Then we can make a multiple T image, means this T, then keep another T. Just in a zigzag way, this animal has to go. Radial maze we discuss. Water maze, there is another maze we call as Barron's maze. Barron's maze what? In between we leave the animal, and in these pockets we'll give some rewards. Like you keep the animal fasting for around 12 hours. Then you keep some foods in this part. Okay, then train the animal to come this part for the food. Okay, the animal if come to the right part, indicate that it is having memory. There is a cheese board we can go. We can observe the different area. This is just modification of this. We can see it in the whole board. Below this, you keep some areas, keep some food. Like this, you kept here in the whole board you keep. So the animal will come here, it will sniff and it will drop its head. So if it go to the right one, you give him the score 1. Wrong one, you give him the score 0. You can observe it here. There is a object localization test or object test. Keep 2 similar objects. One area, one inside a box, you keep food. Another inside, you keep sand. So see how many times rat is going to the sand part or the food part, that you can see. This is a type, we call it complex alley, or we can call it a rectangular maze. What happened here? There will be entry point, there will be exit point. You will leave your animal here. Now it will be zigzag way. The animal has to come in the shortest time. Okay, that time we can observe. We can go for repeated acquisition test. Here the animal has to stay in a particular part. That will allow them. This is called as operant response system. It will come here. Then if you are giving a grid shock, it has to go back to another area. Suppose you keep a box here where you are giving a shock, the animal will jump over here like your uphill. Okay, so these are the various methods by which we can screen the nootropic agents. Thank you all. If anyone has got to ask anything, please. Without any hesitation, you can ask on the blind screenings, or if you want to ask on any screening of any CNS drug, you can ask me.
Students and faculty members, those who have any doubts regarding this drug screening methods on CNS, you can ask to the resource person, Dr. Tripathi sir. Otherwise, you can post it in the chat box also.
Okay, then I don't think anyone has got any doubt. Good. Okay.
Your presentation style is very good, sir. It's as I am at the pharmaceutics department, may also understood very concept is very good, sir.
Actually, Niraja has told me that the listeners will be PharmD students and final year B pharmacy students.
Yeah, sir.
So based on that, I thought that, okay, let me make it very simplified way so that at least they will grasp it. And in your college also, if you want, you can do these small things. If they want some this screening, some smaller one, we can go for it without any much big instruments. We can easily go for analytics, depressant, psychotropics. You can screen it up.
Okay. Today we have the more than 400 participants, sir, from the B Pharmacy and PharmD students also, and some other outside faculty members and students is there, sir. but our Zoom capacity is the 100. Other members is connected with YouTube link, sir.
Okay, so see if there, if there anybody have to ask anything or any queries are there, I can clarify it. I am here for some time.
Okay, sir. Maybe they don't have any doubts, sir.
Okay, good. Fine. Okay.
Thank you, sir.
Welcome.
Thank you very much, sir, for accepting our invitation. We all are happy, sir, with you as a today's resource person in this 2 days webinar. Hope this talk will motivate our students and faculty members to participate in new pharmacological research, and everyone participated in this webinar gain the knowledge on screening of drugs acting on CNS, and also impressed with your presentation style, sir. It was great pleasure to listen to your lecture, sir. Thank you very much for accepting our invitation and given this opportunity, sir. Thank you very much.
Thanks everybody. Thanks Principal Madam, Jairam sir, Neeraj and everybody. I don't know other persons at least. Thank you for giving me this opportunity to tell the students. Thank you.
Thank you, sir.
Okay.
Now we can move to third talk, and I would like to invite Dr. Wani Garu to share her knowledge on trends in molecular biology. And I request Dr. Mrs. K. Sunita to introduce resource person Dr. Wani Garu.
Thank you, sir. Good afternoon, everyone. I, K. Sunita, working as Assistant Professor in Department of Pharmaceutical Chemistry, Vijaya Institute of Pharmaceutical Sciences for Women, Vijayawada. First of all, my sincere thanks to our Principal Madam for giving me this opportunity, and I am very delighted to introduce the profile of our Professor, Dr. Mamilapalli Wani Ma'am, MPharm PhD, who is currently working in the Department of Pharmaceutical Chemistry and Phytochemistry, Vijaya Institute of Pharmaceutical Sciences for Women, Vijayawada. Dr. Wani Madam completed her B Pharmacy from SVE Trust College of Pharmacy, Humnabad, Karnataka in the year 1997. Completed M Pharmacy from Gayatri College of Pharmacy, Orissa in the year 2009. Madam completed her PhD from JNTU Kakinada in the year 2022. Madam has 16 years of teaching and 18 years of research experience. Madam has completed various add-on course programs on patent drafting for beginners from Swayam, regulatory affairs from Raipur Anantapur, research methodology and IPR. Madam has research interest on pharmacognostical, phytochemical and pharmacological screening of crude drugs of herbal origin, phytochemistry and standardization of herbal formulation, design and development of newer drugs from natural products, and Madam has guided UG and PG students. Madam published 36 scientific review and research articles in various indexed national and international journals and peer reviewer too in reputed scientific journals. Madam has patent publication filed on anti-asthmatic and hepatoprotective activities of campesterol. Madam presented and guided for conference presentations and won prizes, and Madam participated in various national and international conferences. Madam published various books and book chapters and delivered various guest lectures. Madam received various awards like Dr. APJ Abdul Kalam Lifetime Achievement Award in the field of teaching, research, and publications from Christ Foundation, Bangalore, in the year 2018. Received Best Research Award from Association of Pharma Professionals in the year 2021. Madam received Best Service Award from VIPW in the year 2022. Received INSO Service Excellence Award from VD Good Professional Association India in the year 2023 and a life member of Association of Pharmaceutical Teachers of India APTI, Indian Pharmaceutical Association IPA, Association of Pharmacy Professionals APP. With this brief introduction, I am elated to ask Professor Dr. M. Wani Madam to deliver the lecture on Trends in Molecular Biology. Thank you, Madam.
Thank you, Madam Sunita. Good afternoon, one and all. I feel myself fortunate to share the session with distinguished speakers like Sabrina sir and Shyamalendra Tripathi sir. And now let me begin the session, which is titled Trends in Molecular Biology. Today I will discuss the introduction, techniques in molecular biology, followed by natural compounds as bioactive compounds. Coming to the introductory session. So molecular biology, it is a branch of biology So it's a branch of biology where we study the molecular basis of biological activity that involves biomolecular synthesis, modification, mechanisms and interactions which are happening in the cell environment. So here we will also study the chemical and physical structure of various biological macromolecules. In this session, we will discuss the interactions between various cell components like DNA, RNA, and protein synthesis and how their interactions are regulated. Molecular biology involves techniques. These techniques are advanced techniques, and these will help us to target new drugs in the drug discovery process. We can diagnose diseases like we have coronavirus spreading time. We have diagnosed with the disease and virus replication pattern, etc. And we can better understand the cell physiology using these techniques. So in medicine, use of this branch is called as molecular medicine. This is a combination or involvement of two subjects, mainly biochemistry and genetics. So in genetics, we'll call it as gene therapy, where genes are manipulated, altering the biological properties of living cells. So genes are expressed, and we'll study the protein, what is synthesized, which DNA is involved. So that is genetics. So we all know cell, which is the basic functional unit of life. Inside the cell, we have nucleus where we find chromosomes. So in these chromosomes, DNA is present. Okay, a part of DNA, a segment of DNA is called as gene. Like this, several chromosomes are present. Several chromosomes are present, and this is called genome. So 23 pairs of chromosomes, they consist of DNA set of instructions. So these terms we should know. Part of DNA is called as gene. So now see, if you look into the structure of DNA, we have two strands. One strand is called axon, and another strand is intron. If you look into the picture, some part of the DNA is coded, some part it is not. Yes, sir. Some part of the DNA is coded and some part is as it is. So the coded one contains the information, whereas non-coded one, it is not having any information. That is, you see, the coded part exon, intron, coded part exon, intron. So like that, DNA, two strands, from the two strands, one strand is separated. That is what we say in the picture as RNA. So this process is called transcription. That means code is transferred from DNA to RNA in the form of one strand. This strand contains both coded and non-coded portions. And in the further step, messenger RNA will call it. So this will contain only the coded information. All the exon parts are separated out here. Okay, so transcription, first step. And next step is translation. Then protein is synthesized. This is called, we say, phenotype. So this is the process. This is the dogma of cell, where this process happens for everything. Like this information is passed. So in the disease process also, this is the thing happening. Some part of DNA, it is sending some messages. So some part of DNA is involved. So what is happening? Those things we will study in molecular biology. So this particular process is called expression. What type of protein is produced in particular disease condition? So this is the basis. Okay, here it involves two steps. In the first step, transcription. Second step is translation, where genotype from the RNA, that is genotype, phenotype, that is protein is produced. Okay, so here we have vaccines. Based on this, we will develop vaccines. A traditional vaccine, generally it is given to body. A pathogen is introduced, dead or alive. Then antigens are produced. Here in case of mRNA vaccines, what we have seen in coronavirus, COVID-19 vaccine is developed, which is an mRNA vaccine. Okay, so here, when you take the vaccine, here mRNA will tell the body, will pass the instructions to produce the pathogens, antigens. Like this will work. As I told you, molecular biology is a junction or combination of two subjects, that is biochemistry and genetics. So biochemistry deals with molecules such as proteins, lipids, carbohydrates, nucleic acids, their synthesis, etc. Whereas genetics, how these genes interactions and expression of phenotype, that particular process is involved in genetics. So in molecular biology, we combining these two, we will study the molecular synthesis, modifications, mechanisms. So your modifications, mechanisms, and interactions. These things we will study, which are, and particularly we use this in studying the disease process. So now let us talk about techniques of modern molecular biology. So first one is polymerase chain reaction, PCR, gel electrophoresis. Here we will use to analyze. Next, second technique is cloning. Third one is blotting and probing. And fourth one is DNA microarrays. There are also other techniques. These are some of the important techniques. So first one, PCR, which is called polymerase chain reaction. So it is a very extremely used technique. The purpose of it is copying DNA. See, here we will produce multiples of DNA. Multiples of DNA is produced here. in using this technique. So primer, the word primer, that means a particular sequence. We know DNA strand contains a sequence of amino acids. Okay, so in the synthesis of this DNA, it will use the particular code. So from this code, multiples are produced in the body. Generally, it will happen. So that particular sequence, which is unique to the DNA, that is called as primer. Okay, so a specific primer here we will use. Okay, next we will use some enzyme that is called polymerase enzyme. So this amplification process needs enzyme called polymerase. In this, with this only we can amplify. Okay, so from DNA to RNA, and we'll get the protein. That is the process generally. So reverse transcriptase means from RNA to DNA. So which RNA and produced from what type of DNA? So what is the DNA? What is the sequence of DNA? So that is called reverse transcription PCR. So how much amount of this RNA or DNA is present? That also we will find out using qPCR, that is quantitative PCR. See, this is the process. We have taken the required DNA sample. It is having 2 strands, blue and black. Okay, so whichever the area we want to copy, multiples of that copy, we have selected that part. Okay, next, we have first heated to 95 degrees. At that time, the 2 strands will get separated. Next, we will cool to 55 to 60 degrees. At this time juncture, we add primers. Primers are specific code they are having. So these primers, they are going to attach at particular place to each of the separated strand. Next, it is heated to 72 degrees. At this place, we will be adding enzyme, that is polymerase enzyme. Okay, and with this addition of polymerase enzyme, see, this primer is getting enlarged. So that means a new DNA strand is formed, black and orange. Another orange and blue. So earlier we have only one DNA with the 2 strands. Now at this place we have 4 strands which contains 2 DNA molecules. Okay, with that specific code or particular region with sequence. So we have that multiple numbers. Okay, same thing again, you heat to 95 degrees. Again, these will separate, again add primers. This time 4 and the next time you will get 8, like that, double we will get. So this particular, see this here I am showing, primer will go and attach at the place where its complementary strand is there. Okay, it will identify. So this is the DNA sample. We require some primers, nucleotides, polymerase enzyme, buffer, and we require PCR tube, like just like centrifuge tube. In this, we will add all these things. We'll take a PCR cycler, and we will give, we will set the same temperature. We are just changing the temperature. At that time, all these things will happen. First step, separation of strands, that is called denaturing. Next, we are adding primers. It is called annealing step. Next step is called extension, where we will get multiples of DNA. So this is PCR. So next, how many DNA are produced, whether they are different or same, what type of sequence is present, we want to analyze. Okay, how? For that, we use a technique called gel electrophoresis. Okay, here we separate the molecules depending upon their size. Here we use gel, which is made of agarose or polyacrylamide. This is one of the important tools used in molecular biology. The DNA fragments are here separated. We are getting number of DNA fragments all in a group, and we will separate into individual. Just like TLC, we will separate the components. In the same way here, we will separate the fragments of DNA. So in this gel electrophoresis, a gel-like substance is poured, and it will have 2 electrodes, positive end and negative end. So this DNA molecule backbone will move towards the negatively charged end, phosphate groups. The DNA will migrate towards the positive end, like that. So here we will, the types of gels we use, SDS phase gel, and electric charge we can apply using 2D gel electrophores. This agar gel in borate buffer. Next, SDS page, another. So sodium gludecyl sulfate polyacrylamide gel. So this is the equipment, and the separated DNA spots will be like this. So in crime branch also it is used. For example, you have collected hair of any victim. Okay, that is placed into this PCR tube, and it is subjected to PCR, where it will multiply and number of DNA strands will be separated. We will separate them, and we will use this gel electrophoresis technique. Okay, we will inject the sample in the place where it is needed to be placed, and we will run. Okay, so and you see separation of this DNA, whose DNA is. So now you can identification of DNA. For this technique also we can use. For that purpose also we use this PCR, whose DNA. Okay, so the lane 1 marker, lane 2 sample from the crime scene, lane 3 is of victim DNA, lane 4 suspect 1, lane 5 suspect 2, lane 6 suspect 3, like this the samples. From the samples we can separate the DNA and we can analyze. In various fields we can use. Next technique is DNA, gene expression or cloning. So the process of generating genetically identical copy. So genetically identical copy of a cell, it is called as cloning. Generally in nature it will be happening. In prokaryotic cell, bacteria automatically create their duplicates by binary fusion or budding. In eukaryotes we see cells undergo mitosis, like our skin cells, cells underlining the GI tract. They are called clones. So a exception is meiosis, where there is genetic recombination, a change, eggs, sperm, some fertilization process has to be involved. So changes will be there. But in case of mitosis, same identical genetic material will be copied and reproduced. So this technique is used to understand the protein function. So particular protein is here cloned or copied using PCR. Again, we use this technique of PCR. So plasmids. So these plasmids are present in bacteria. They are round-shaped chromosomes. Okay, that particular DNA, which is code for protein. Earlier I told. So DNA, RNA, protein. Okay, so protein is produced means from which DNA it is produced. So that will be having some code. So that we have to identify. If you want to study the disease pattern, we have to produce it in multiple number and we will study. Okay, if you want to transfer it, that particular coded DNA, we will transfer it to plasmids, bacteria, and we will study. Okay, animal cell or bacterial cell. This will be helpful in case of antibiotic resistance, antibiotic studies, like that. So reproductive cloning. So here what we'll be doing? So cloned embryo will be implanting into uterus. That will develop into fetus. So here it is developed by using cells like stem cells, somatic cells, gamete cells. So stem cell technology, it is also cloning. And we'll say molecular cloning, that is what I said, DNA coding, how a protein is expressed, from which DNA it is expressed, what are the various signaling factors that help to express a protein. Okay, those all things. What is the enzymatic activity? So what is the tertiary structure of the protein? By crystallizing it, we can study. So this point, particularly pharmaceutical industry uses, where they will study the activities of new drugs in research. Next, third one is blotting techniques. Here, these techniques are used to detect and analyze 3 types of macromolecules: DNA, RNA, and proteins. Okay, so there are 3 methods: Western blot analysis for analysis of proteins, Southern blot analysis for the analysis of DNA, Northern blot analysis for the analysis of RNA. Here also we will use gel electrophoresis, and we will use a nitrocellulose sheet to analyze, and by autoradiography, by using some fluorescence, we will study what kind of samples, how much amount they are present. So this northern blot technique, you see, we have taken the DNA molecule, we have cut it into fragments. We have taken the agarose gel. We have placed the fragments where they are required to be placed. And next we have transferred into a membrane, that is nitrocellulose filter. See, on this filter, those fragments are there. Now here we will apply some probes. So earlier I told primers, and these are probes. So primer means that will be unique to a particular DNA. Using that sequence, the next replication will start. Probes are in the DNA strand, some particular kind of sequences will be repeated. So that kind of strands, that kind of particular code, those are called probes. So they are unique to that particular DNA and they will go and attach to a complementary strand. So here they will take those probes. So these DNA fragments will unite with the probes by using fluorescence, and we will study using X-ray films or autoradiography. This is northern blotting for DNA analysis. For RNA analysis, in the same way, we use this southern blotting technique. RNA is extracted. Okay, again we will apply to it gel electrophoresis. We use some membrane, that is nitrocellulose sheet. Again, probes, labeled probes here. See, radio, fluorescence, labeled probes we use, and they will unite with that. And what type of component it is, that we will analyze from the intensity produced using this radio autograph instrument. Next, DNA microarrays. So here arrays are some solid supports made of glass or silica, where we will be attaching the DNA. So many number of DNA are attached in a grid fashion. Small round blocks of a plate are made, in which each plate contains this gene. Okay, each part of DNA is called as probe and it represents a single gene. So with this we can analyze tens and thousands of genes simultaneously. Okay, so these are called DNA chips, DNA arrays, gene arrays, biochips, like that. So nucleic acid strands, what type of nucleic acid strands, what is the difference, what type of DNA it is, like this we can analyze like that. Number of DNA molecules can be analyzed in a less time. Next, the property, as I said, complementary nucleic acid sequences, they unite with each other by forming hydrogen bonds. Here also we will use the labeled probes or fluorescent dyes. So these labeled target sequences bind to a probe sequence. Here we will use a diseased and normal DNA and diseased DNA. So what is happening? I will see and understand. So the procedure will be, we have to collect the samples, isolate the mRNA. Next, you will create a labeled seed in a complementary DNA strand. Next, you hybridize, we will collect and analyze. You see, this is the healthy cell and pathological cell. We have taken the cell, we have cultured the cells, we have isolated the RNA. By reverse transcription and fluorescence tagging, we have used the probes and we will identify. Okay, like this we will get red, yellow, green, and black. So normal cells, pathological cells present in both cells. So in this way, we will understand. So particularly in cancer cell studies, cancer cell lines, we will use this technique. So we will identify the genes responsible for a specific disease. So this is used in gene targeting and gene therapy. This is called. So now let us look at some of the natural products, their molecular mechanisms. So apigenin, it is a hydroxyflavone compound present in some herbal extracts. It is used to treat xerostomia. Xerostomia is a condition often occurs in menopausal women where there is reduction in salivary production. The salivary glands will be producing less amount of saliva. Because of that, mouth dryness will be there, burning sensation will be there, and difficulty in swallowing. That type of problems will be faced. So in vitro studies have been conducted using these human salivary gland cells and estradiol. In vivo studies were also conducted ovarioectomized mice model. So here they found that the mechanism is apigenin upregulates aquaporin 5. So aquaporin 5, it is interacting with aquaporin 5 at the molecular level or in the cell. Okay, so restoration of saliva flow rates happen by the usage of this apigenin. Okay, so here one more mechanism involved is ER alpha signaling pathway. So by this way it works. Apigenin, it is a newer molecule. Still further studies are required. Next, beta-cetosterol, it is a plant-derived sterol useful in angiogenesis and useful in treatment of rheumatoid arthritis. So studies are conducted both in vitro using human umbilical vein endothelial cells and in vivo using collagen-induced arthritis mice models. Okay, so they found that here cetosterol inhibits VEGF signaling. So this interacts with VEGF signaling. Okay, so it decreases cell proliferation and migration. So the swelling degree and the damage of bone and cartilage, synovial angiogenesis inhibition, and reduced the expression of VEGF-1/2. So it reduced that expression. So this data will help us that cetosterol is having anti-VEGF. Next, levistilide. It is a phthalate derivative. It expands human functional umbilical cord blood stem cells. Okay, it has increased. It was tested in vivo and it has shown rapid engraftment of stem cells. So this may be due to reduction of reactive oxygen species, ROS. Next, alpha mangosteen, another molecule from mangosteen hull. In vitro assays in different cancer cell lines showed that it increased apoptotic mechanisms including PARP cleavage, BAX induction, and blockade of AKT signaling. So in this way, alpha mangosteen works as anti-cancer agent. Like berberin and berberibin. So berberibin is a metabolite of berberin. These compounds inhibit RASA3 RAP1 pathway. It is critical for ADP-induced integrin activation. Okay, in thrombosis, carrageenan-induced thrombosis in mouse tail, the antiplatelet effect was studied. Next, vartioxetin or fluoxetine. It prevented oxidative stress, so it is used in neurological disorders. It prevented oxidative stress in the hippocampus of A-beta injected mice. It decreased inflammatory markers, nitric oxide synthase and NADPH oxidase 2. It increased glutathione peroxidase 1, an antioxidant enzyme. It offers neuroprotection, and it was confirmed in vitro in neuronal cultures challenged with A-beta oligomers. So this is used in Alzheimer's disease, which is a depression-related disorder. Next, phenolic lipids here, especially resorcinolic lipids are also
వస్తుంది మీ దాంట్లో
Sorry for the interruption. So phenolic lipids, especially in this lipids, we'll study or we'll talk about resource-nolic lipids as bioactive compounds. They are isoprenoic compounds related to the phenol or dihydroxyphenol. They are derivatives of dihydroxybenzene. We know in this class catechol, hydroquinone, but they are all well known. Resorcinol derivatives are of interest. The phenolic lipids are lipids because they consist of a phenolic structure with a long aliphatic hydrocarbon chain. They are hydrophobic molecules. They are present in Anacardiaceae family, cashew, mango, the Ginkgoaceae family, Ginkgo biloba, and gramineous cereals, rye, wheat, barley, and rice. They are amphiphilic compounds with octanol water partition coefficient log P ranging from 7 to 12. Therefore, with this, we can say they interact with biological membranes and they change or interact with the structure and function. These work with the DNA damage, they prevent their repair, especially useful in anti-cancer agents like bleomycin. So the cereal grains which contain these resource-nolic lipids act as phytopathogenic fungi. They give produce resistance against these phytopathogenic fungi. Next, cellular metabolism therapy by small natural compounds. For metabolic dysfunction, so we have to counteract this problem. This plays a prominent role in the pathophysiology of different diseases. Metabolic dysfunction involved diseases such as cancer, diabetes, metabolic syndrome, cardiovascular, and neurodegenerative diseases. So bioactive chemical compounds are discovered in nature. They are abundantly available. They are sustainable. They have greater tolerance. They act as excellent alternative to synthetic drugs. But the problem is they have there is less knowledge about their metabolic pathways, their capacity to support and modify. And therefore, this problem to be overcome. The metabolic pathways involved to overcome the diseases involved in natural compounds, it must also be studied. Next, so mitochondrial dysfunction is the cause of bioenergetic alteration, triggering pathological conditions, excessive ROS production. In mitochondria, it leads to biomolecular damage in the cell. It is responsible for inflammatory process characterizing human diseases. Plant Xantholina pinnata. This has been recently studied. The extracts have shown very good results in cancer studies. So therefore, we can say that the extracts are having phytochemicals which may be novel anti-cancer and anti-inflammatory. So cardiovascular diseases, atherosclerosis are pathological conditions triggered by metabolic disorders such as dyslipidemia. So the potential of a natural compound picrocrosin, it has been investigated in HEP G2 cells. It is having hypolipidic properties. It is extracted from saffron stigmas. It exerts reduction of triglyceride synthesis and increase in LDL reuptake through non-statin-like mechanism. Hyaluronic acid. This is also one of the attracting compound. HA-based products are used in skin problems, cancer therapy, inflammatory disorders. Next, another novel glycolytic inhibitor, natin-H. It controls cellular glucose homeostasis, melanoma, and cancer, anti-cancer drug. It is used along with fenfarmine. Next, another anti-cancer agent, doxorubicin, bergamot polyphenolic fraction. They all interact with mitochondrial parameters of oxidative metabolism of PAECs impaired by DOX, and they're reboosted with the use of PPF. So with this, I come to the conclusion. The interactions at the molecular level between chemical mediators and cellular systems arise from biochemistry and molecular biology studies, and they provide the basis of drug therapy and represent a useful and fundamental tool for developing new therapeutic approaches that are more efficient and ensure an improved quality of life. So natural mediators of cellular metabolism, both endogenous and exogenous, derived from plants and animal sources, become an intriguing research issue that needs to be expanded upon. Thank you. Thank you for giving me this opportunity. I thank Principal Madam and all of you for paying attention to me. If you have any queries, you can mail me or you can ask me. Thank you.
Thank you, madam. On behalf of faculty and IQAC VAPW, I would like to thank you very much, madam, for active participation in this 2 days webinar. Thank you for your wonderful talk on recent trends in molecular biology. And you have started from this talk with beginners of point view and explained it with various examples. After this talk, we have confidence on molecular biology, madam. Thank you very much for giving this opportunity. Now, I request Mr. A. Jairam Reddy Garu to end this first day session with a vote of thanks.
Good afternoon, everybody. This is Jairam Reddy from Department of Pharmacology, and today we are conducted a 2-day national level webinar on the topic of recent horizons in experimental pharmacology and molecular biology. So first and foremost, I would like to express my appreciation and special thanks to the speakers for their valuable contribution to our 2 days national webinar on recent horizons in experimental pharmacology and experimental pharmacology and molecular biology. It is mandate to oversee the implementation of the aims to keep the students need to update the latest development in research industry. So by hearing and learning from the speakers, it has been educated me along with our students and participants. My deepest gratitude to those all who attended the webinar and helped make it such a successful event. So first of all, I would like to give the highlights of this first day webinar. So Dr. Sabrina sir has explained about the cellular compositions which are in the human body, which are involved in the various inflammatory mechanisms by various signaling pathways, both by the exogenous as well as the endogenous. So then, along with that, sir has explained the drugs which are going to use in the inflammatory diseases by using the COX-1 inhibitors as well as the COX-2 inhibitors. And along with that, the advanced therapies by using like the steroidal agents as well as the inflammation mediated diseases like arthritis can also treated by using that is monoclonal antibodies. So that can able to change the gene expression and which can able to prevent the inflammatory diseases. And along with that, the sir also explained the few of the models which can able to use for the determination of the anti-arthritic diseases, the mediated diseases like arthritis. So then the second talk by the Dr. S. Tripathi from Shikshavo Anusandhan University. Shikshavo Anusandhan University. Sir has explained about the different types of screening methods. So that is the basic methods what we are going to use in both preclinical as well as the clinical level also. So along with that, the sir has explained about the nootropic agents. So which are the recent, which is drugs used in the treatment of forgetfulness, especially the most of the conditions because of the radiation as well as the various environmental conditions as well as the lifestyle. So the most of the population is getting this type of diseases. To prove the new or existing drugs by using the various animal models as well as the techniques to determine the nootropic agents. And sir also explained the possibilities of the nootropic evaluations. So then sir has explained the new molecular techniques, or either it may be the experimental animal models or experimental procedures as explained in detail. So then the third talk has been given by the Dr. M. Wani, professor from the Vijay Institute of Pharmaceutical Sciences, explained about the recent trends in molecular biology. So here the madam has explained the modern molecular techniques, so which can able to use to determine the various diseases as well as the new drugs from the both synthetic as well as the natural RSN. So Sarah madam also explained with some of the examples and which are going to use in the cell proliferation inhibitors as well as the metabolic disorders. So then so all the speakers as highlighted the experimental models and the techniques used in used in the various diseases that helps the students for their research work. So before ending my closing remarks, I would like to convey my special thanks to the speakers who have been extremely cooperative and staying with us beyond the time allocated. So finally, my deepest thanks to the participants as well as my colleagues and the students who are participated in the national webinar. Thank you, one and all.
Thank you, Jairam, sir. Thank you all for your active participation. Thanks to our faculty members and beloved principal madam for your support to organize this 2 days webinar. Thank you all for giving this